DVD3 will begin with a discussion of milieu therapy with vitamin D and fish oil. Let's first examine the effects of vitamin D on differentiation and activity of dendritic cells, those are antigen-presenting cells, and T lymphocytes. Can we influence the differentiation of T cells to go towards non-inflammatory Th2 or regulatory Treg and away from pro-atherosclerotic, pro-heart failure Th1 and Th17? Let's take a look at this. Now again, dendritic cells, antigen-presenting cells, are the first line of our innate defense mechanisms. These cells consist of monocytes in the blood or the spleen, macrophages in our tissues, Langerhans cells in the skin, or Kupffer cells in the liver. These dendritic cells have patterned receptors on their cell membrane to recognize invaders. They will recognize and engulf the invader, digest it, hightail it to the nearest lymph tissue, and on their way, they will convert from antigen capture cells to antigen presenting cells. They will arm themselves with co-stimulatory molecules such as B17, and they will then present an antigenic determinant to a non-committed or resting T cell. And if they present co-stimulatory molecules, they will cause that T cell to activate. If they do not present co-stimulatory molecules and they simply present an antigenic determinant to the resting T cell, there will be an immune non-event. The T cell will not activate, it will actually become anergic. If they are displaying inflammatory co-stimulatory molecules, then the T cell will activate, typically into a Th1 or a Th17 pathway, which promotes atherosclerosis and heart failure. However, if the antigen-presenting cell is presenting regulatory or suppressive molecules, that will encourage the T cell to differentiate into a T regulatory cell, which will quiet down abnormal immune responses. Will vitamin D push T cells towards a T regulatory and away from a Th1, Th17 response? Let's take a look. Now again, the initial decision, are we going to be a Th1 or a Th2 cell, is related to characteristics of the invader. If the antigen capture cell, the dendritic cell, thinks that the invader is a microbe that macrophages will kill in an intracellular fashion, then the antigen presenting cell will release interleukin 12 and thus will get a Th1 response. If the antigen capture cell, the dendritic cell, thinks that this is an invader that we're going to kill with antibodies such as a parasite, it will release interleukin 4 and we get a Th2 cell. So this decision, Th1 versus Th2, is in part related to the perceived characteristics of the invader. Now, in the situation of atherosclerosis and heart failure, the perceived invaders, oxidized LDL or troponin I or other altered endogenous proteins, will be perceived as an intracellular organism. We would tend to have a Th1 response. Above and beyond the characteristics of the invader or the perceived invader will be the cytokine milieu in which these immune interactions are occurring. If the immune interaction is occurring in a milieu rich in Th1 cytokines such as interleukin 12, that will coerce the T cell to go Th1. It will make interferon gamma and other Th1 cytokines. That's deleterious from our point of view. If there is a lot of interleukin 4, it goes to Th2, which is benign from our point of view. If there's interleukin 6 and interleukin 23, two bad guys, we go Th17. That's a problem for us in heart failure atherosclerosis. However, if we have interleukin 6 and some TGF-beta, that's transforming growth factor beta, a regulatory cytokine, that T cell will tend to commit to Treg. Treg cells make interleukin 10 and transforming growth factor beta, which are regulatory, which will quiet down the immune response. We want a lot of Tregs in our circulation, in our arteries, and in our hearts. Let's look at the effects of these regulatory anti-inflammatory cytokines. Let's start with transforming growth factor beta. Let's look at its role in plaque stability. We're going to take 24 patients scheduled for carotid endarterectomy, and they all have an at least 70% narrowing. They're all on aspirin. Now, 12 have asymptomatic disease. Their carotid ultrasound was abnormal. They were not having symptoms. 12 have recent onset symptoms. Therefore, we know that the immune system activated within the plaque. There was a plaque ulceration or a plaque fissure. We know this represents an immune frenzy within the shoulder region of that plaque. Now, they had similar percent narrowing, similar risk factors. One group had active disease, one group had stable disease. Now, they both had, on average, 76% narrowings, but those with symptoms, with plaque activation, they were much more likely to have plaque ulceration, and that's what caused their symptoms to develop. Now, if we look at the microscopic characteristics of the plaque, in the patients who are symptomatic with active plaques, you can see they have fewer smooth muscle cells, more macrophages, more antigen-presenting cells, demonstrating major histocompatibility to molecules upon which they're presenting antigenic determinants, revving up this immune response. And as you would expect, they have a greater representation of T cells within their plaques. And again, we know that activation of the plaque involves a loss of the fibrous cap, a loss of smooth muscles related to the effects of interferon gamma and matrix metalloproteinases. That's what activates plaque. A plaque rupture or plaque activation represents immune frenzy, loss of regulatory control. Now, if we look at the percentage of the plaque containing the regulatory suppressive cytokine transforming growth factor beta, the symptomatic patients have less TGF-beta, as you would expect. The TGF-beta content in the asymptomatic plaque is greater. Now, is the TGF-beta being made in the plaque, or is it entering the plaque from the circulation? If we look at messenger RNA, which is a product of gene transcription of the immune cells, we can see that the immune cells within the plaque of an asymptomatic patient are making more TGF-beta. So this inflammation or regulatory activity is occurring within the plaque, not the circulation. If we look at collagen, we want a lot of collagen in our plaque so we have a resilient fibrous cap to the plaque. We see much more collagen in the stable plaques than in the unstable plaques because in the absence of TGF-beta, we have unrestrained Th1, Th17 activity. We're going to chew up our collagen and our smooth muscles. That's what makes the plaque vulnerable. That's why they had symptoms. So TGF-beta restrains immune activity and maintains stability of plaque. What about the other Treg cytokine, interleukin 10? In this study, we're going to look at female mice, wild-type mice that can make IL-10, and then genetically abnormal double knockout mice, and that they cannot make IL-10. They're otherwise the same, except one population of mice can make IL-10, the other mice cannot. You're going to put them all in the same atherogenic diet. In this mouse model, you're going to develop some plaque. You'll house them under different conditions. One group is in standard lab conditions, being exposed to the bacteria and viruses that mice and humans would experience in everyday life. Every day we're exposed to microbes, and we will mount an immune response. We will kill the microbes, and then the immune system will stand down. Another group was housed in microbe-free aseptic conditions, kind of like being in a bubble, so you're not going to be exposed to any viruses or bacteria. And then you'll sacrifice the animals and look at plaque formation. Later on, some of the IL-10 negative mice will be treated with IL-10. You infect them with a plasmid, which is exogenous DNA, kind of like a virus, to see what happens if you reconstitute the IL-10 that they cannot make on their own. Now, this is a picture of the aortic root. This is the aortic valve. And in red here, you see plaque. The wild-type mice that could make IL-10, they had some plaque, and it didn't really matter whether they were in the bubble environment or the standard laboratory conditions where they were exposed to the usual bacteria and viruses that humans and animals would experience. Now, the IL-10 knockout mice who were unable to restrain their immune response. In response to infection, we rev up and then we quiet down. Without IL-10, we have trouble quieting down. So the IL-10 knockout mice in the bubble aseptic conditions had a lot more plaque than the animals that could make IL-10. That makes sense. Now, what's interesting, the animals housed in standard laboratory conditions where they would be exposed to the infections of everyday life had a lot more plaque. Each time we are exposed to infection, the immune system revs and then we quiet it down. If we don't have T regulatory function, we don't have IL-10, we don't quiet down. We have kind of a chronic inflammatory response. Chronic inflammation alters the LDL, causing it to be trapped such that free radicals can oxidize it. So without IL-10, without the ability to regulate or quiet down the immune response, we respond to infections with ongoing inflammation. So we want a lot of IL-10, we want a lot of TGF-beta, we want high levels of Treg activity. In the IL-10 negative mice, they had more T lymphocytes, much more interferon gamma, Th1 cytokine, much less collagen because they were chewing it up. Now, if you look at the plaque area of the IL-10 knockout mice, they have a lot more plaque. If you infect them with the exogenous DNA to make IL-10, they have much less plaque. So the key here was IL-10, a T regulatory cytokine. So we want our immune system to be in balance. Because we are overweight, because we don't exercise, because we are loaded with heavy metals, because our diets are poor in antioxidant minerals, we Americans are in an imbalanced situation with too much Th1 and too much Th17. We want to change our milieu and get back in balance with more Treg activity. Let's look at the effect of vitamin D supplementation on a Th1, Th17 upregulation condition, congestive heart failure. We're going to take 93 patients with congestive heart failure. We'll carry out baseline measurements and randomize them to receive over 9 months vitamin D, not a high dose, 2,000 international units daily with 500 milligrams of calcium. The control group receives calcium without vitamin D. All standard therapies continued. You'll reevaluate at 9 months. A double-blind protocol was followed. Vitamin D levels did not rise significantly with placebo therapy, nor would you expect that, but 25 hydroxyvitamin D rose significantly with supplementation. This is the vitamin D level that we follow in the blood. The active form of vitamin D is calcitriol, 1-hydroxy, 25-hydroxyvitamin D. Most cells of our body, including immune cells, can convert the vitamin D that you receive as a supplement to the active calcitriol. Calcium levels and parathyroid hormone levels did not change significantly. Now, if you are severely deficient in vitamin D, your calcium level will be low. The parathyroid gland will release parathyroid hormone to remove calcium from bone to raise your blood calcium level. And parathyroid hormone is toxic to the heart. Now, these patients were not severely deficient in D or calcium. We're not affecting parathyroid hormone dynamics. We're just replacing vitamin D. And again, we want to have a vitamin D level of at least 30. These patients were low at 14, and we increased them to 41. So we raised their vitamin D level. Tumor necrosis factor alpha increased over 9 months with placebo therapy. That's not a surprise. Heart failure is a Th1, Th17 condition. It fell a little bit with vitamin D. C-reactive protein was unchanged. Not every Th1 cytokine will move in tandem with different therapies. But interleukin-10, the Treg cytokine that quiets things down, fell over 9 months with placebo therapy, as we would expect, and it rose with vitamin D. That's a very positive sign because vitamin D will raise regulatory cytokines that will quiet down the immune component of your heart failure and atherosclerosis. Now, we talked about the factors that determine the pathway that a T cell will follow. This has to do with the perceived characteristics of the invader. It also has to do with the cytokine milieu. The dendritic cell, the characteristics of the antigen-presenting cell, may also determine which pathway the T cell will follow. Let's see if vitamin D can affect the characteristics of the dendritic cell such that the immune bias will move away from inflammation and more towards T regulatory activity. Now again, these dendritic cells, the antigen-presenting cells, convert from an antigen capture cell to an antigen-presenting cell and then will present their antigenic determinant to a T cell with co-stimulatory molecules. A mature, activated dendritic cell will present B7 and other pro-inflammatory co-stimulatory molecules such that the T cell wants to go Th1, Th17. An immature dendritic cell will not display co-stimulatory molecules. As a matter of fact, it will display co-stimulatory molecules that will coerce the T cell to go into a Treg pathway. Vitamin D arrests the differentiation of dendritic cells such that they tend to encourage T cells to take the Treg route. Let's examine this in an animal model of Crohn's disease. We're going to demonstrate that vitamin D promotes a shift from Th1 to Th2 and Treg. We're going to look at 8-week-old biopsy mice. We're going to treat them with retention enema containing a molecule called TNBS. This will damage the bowel wall and will lead to Crohn's disease, and this is a mouse model, so we'll call it murine Crohn's disease. We feel that Crohn's disease, we know it's not an immune condition, the immune system is misrecognizing either the normal bacterial flora of the colon as an invader, tolerance was lost, or the bowel wall has been altered such that it is perceived as a threat. Now, so they're all going to have Crohn's disease, the murine model of Crohn's disease, and then you're going to treat them with intraperitoneal calcitriol, that's the active form of vitamin D, and or dexamethasone, that is a glucocorticoid. The standard medical therapy for autoimmune disease is immune suppression with exogenous corticosteroids such as decadron or prednisone. So the animals are treated either with vitamin D and or a prednisone type therapy. 2 hours before they receive the enema that will bring on the murine Crohn's disease, and daily for 3 days afterwards. Then you're going to remove their colon at day 3. This is a normal colon with a smooth wall. These 2 little nodules are handling artifact. This is the microscopic appearance. These are the cells that line the bowel wall. They're smooth and symmetrical. This is the basement membrane and the back wall of the bowel. This is the colon of the animal with untreated Crohn's disease. You can see it's not looking so good. 3 and 4 are animals treated with vitamin D alone or steroids alone. They're looking a little bit better, but the animals that receive both vitamin D and the prednisone, they're looking a lot better. That bowel wall looks a lot more normal than the untreated animals. Why? If we look at levels of interferon gamma and tumor necrosis factor in the bowel wall, those are Th1, Th17 cytokines. With the Crohn's disease, they're sky high. The animals that were treated with vitamin D, they're much lower. The animals treated with the prednisone alone, the dexamethasone, are lower. There's a synergy between vitamin D and steroid therapy. As we increase the dose of steroid therapy, the levels of the Th1 and Th17 cytokines fall, and synergy with vitamin D is maintained. Same situation with the other inflammatory cytokine, interferon 1 beta. Myeloperoxidase is a free radical enzyme. It is a marker of free radical stress, similar to finding elevated malondialdehyde in a patient with arterial disease. We have a lot of free radical stress in untreated Crohn's disease. This is attenuated with vitamin D, with steroid therapy, and again, there is a synergy between vitamin D and steroid therapy. T-BET is a Th1 commitment factor, and if 100% of the if in the untreated Crohn's disease, the T-BET expression is 100%, it's decreased by 45% with vitamin D, by 70% with steroid therapy, and by 82% with a combination. So both vitamin D and steroid therapy will downregulate Th1. GATA3 is a Th2 expression marker. It is doubled with vitamin D treatment. steroid therapy doesn't do much, but there's a synergy between D and steroid therapy. It rises fivefold. So both D and steroid therapy will decrease Th1. D, but not steroid therapy, increases Th2, but D and steroid therapy will increase Th2 in a synergistic fashion. Interleukin 10 and transforming growth factor beta, those are Treg cytokines, rise significantly with vitamin D. Dexamethasone, the steroid therapy has less of an effect, but there's synergy between D and dexamethasone in increasing the Treg cytokines. FOXP3, that is a marker of Treg cells, will rise 3.5-fold if you add vitamin D to steroid therapy. CTLA-4 expression, the Treg cells release into the milieu IL-10 and TGF-beta suppress all the immune cells in the region. They also display on their cell membrane, basically a membrane-bound cytokine, CTLA-4, that mediates contact suppression. So if they come across other immune cells, they will ligate receptors on the immune cell with CTLA-4 and quiet things down. So it rises with vitamin D. Steroid therapy had a benefit, and again, there was a synergy. IL-12 is manufactured by dendritic cells to call in resting T cells that want to go Th1. IL-23 is manufactured by dendritic cells, antigen-presenting cells, to call in T cells that want to go 17. They are upregulated in Crohn's disease versus untreated animals, and they are suppressed by vitamin D, by steroid therapy, and again, there's a synergy. So with vitamin D, we're downregulating Th1, Th17, upregulating Th2 and Treg. With steroid therapy, we're pretty much downregulating everything. There's a synergy here. Now let's look at vitamin D interacting against tumor necrosis factor alpha in placental function. Elevated Th1 cytokine release, that's TNF-alpha, interleukin 1 beta, interleukin 6, is associated with a poor outcome of pregnancy, miscarriage, preterm labor, preeclampsia. Consider the placenta. It is an organ of immune privilege. Now, when you're carrying your baby, half of the proteins in your baby are not normal. If you would receive a kidney transplant from your adult child without immunosuppressive therapy, you would reject it because it's abnormal protein. Why don't you reject your baby when you're carrying your baby? It's because the interface between you and the baby, the placenta, is an organ of immune privilege full of T regulatory cells. So it makes sense that if we have inflammatory cytokines and a lot of Th1 and Th17 cells in the placenta, this might compromise the integrity of the placenta and compromise the outcome of pregnancy, and indeed that's the case. Now, the placenta can generate the active form of vitamin D from the supplementary form of vitamin D, and it can also degrade vitamin D. So it can make vitamin D and it can degrade vitamin D. If you are deficient in vitamin D, you have a fivefold relative increase in preeclampsia. That is a hypertensive crisis of pregnancy. In this study, you're going to incubate placental cells, those are called trophoblasts, with varying doses of tumor necrosis factor alpha and calcitriol. Assess the effect on cytokine, enzyme, and hormone production by the placental cells. Now again, tumor necrosis factor alpha will be sensed by receptors on the cell membrane of an immune cell. That will cause nuclear factor kappa beta to translocate to the nucleus, where it will lead to the production of Th1 and Th17 inflammatory cytokines, including tumor necrosis factor alpha. So tumor necrosis factor alpha leads to more tumor necrosis factor alpha. Now, here we're going to look at TNF levels without adding in additional tumor necrosis factor or vitamin D. They're fairly low. If we pour in tumor necrosis factor alpha, we get more TNF out because we're moving nuclear factor kappa beta into the nucleus. If we treat the cells with tumor necrosis factor alpha and increasing doses of vitamin D, TNF production is attenuated. Same situation with interleukin 6 and interferon gamma. So TNF leads to inflammation. that TNF-induced inflammation is attenuated by vitamin D. Tumor necrosis factor alpha leads to an increase in all the Th1, Th17 inflammatory cytokines. This effect is blunted by calcitriol. That's why vitamin D will protect the pregnancy. There's an enzyme, CYP24A1, that degrades vitamin D. The activity of that enzyme is upregulated by increasing doses of TNF. So vitamin D attenuates inflammation. Inflammation attenuates vitamin D. So we need to have a proper balance here. The placenta generates a number of hormones necessary to maintain the viability of the fetus, including estradiol, progesterone, and HCG, human gonadotropin. As you incubate these cells with increasing levels of tumor necrosis factor alpha, the placental function deteriorates. So we don't want a high level of inflammation in the immune-privileged organ, the placenta. We want a high level of vitamin D to maintain tolerance in the placenta. Now, we demonstrated that vitamin D is helpful in a murine model of Crohn's disease, and it makes sense that it would be helpful in humans with Crohn's disease. Let's examine that. 94 patients with Crohn's disease in remission. They were all off steroid medications, and they had a low level of inflammation. They had a Crohn's disease activity index below 150. They're in remission. You do baseline measurements, randomize them to receive over a year 1,200 milligrams of calcium and 1,200 international units of vitamin D, not a high dose, or the control group simply receives calcium and placebo. You're going to monitor for clinical relapse, and a double-blind protocol was followed. Now, vitamin D levels went up with active therapy from 28 to 38. They didn't really change with placebo. Individuals recruited in the winter, so they're moving into the sunny summer. Vitamin D rose in both groups, but it rose more with supplementation. Those recruited in the summer, then in the winter months with less sunlight, the levels fell a lot more with placebo therapy. They didn't fall as much with vitamin D. So we give you vitamin D, the levels go up. How'd the patients do? Relapse rate over 1 year was decreased by over 50%, from 29% to 13%. This is relapse-free outcome. The patients on vitamin D did much better. Fish oil and Crohn's disease. We'll talk about fish oil later, but if vitamin D is helpful in an autoimmune disease by increasing Treg activity, we'll talk later on that fish oil increases Treg activity. We would expect that any disease where vitamin D is helpful, we would see a benefit of fish oil. Let's see if fish oil helps in Crohn's disease. 38 patients with Crohn's disease in remission, randomized them to receive over 2 years a controlled diet or a diet rich in cold water fish that has a lot of fish oil, EPA and DHA. There was relapse in 58% of those on the control diet, 20% on the fish oil diet. Another study, 78 patients with Crohn's disease in remission, randomized them to receive over 1 year enteric-coated fish oil to give them about 2,000 milligrams of EPA and DHA combined or placebo. Remission is maintained in 26% of placebo therapy, 58% in the fish oil. So the fish oil group obviously did quite well with Crohn's disease, same as they did with vitamin D. We want to have a lot of T regulatory cells throughout our body. It's not necessary that we mount a T regulatory response against every antigenic determinant in the artery wall or in the heart. But the more molecules that we mount a Treg response to, the less Th1 and Th17 inflammation we will see. In our animal model, where we created a Treg response to a snippet of troponin I, we saw that having a Treg response to that one molecule lessened the degree of damage in an experimental heart attack. Now, in the experimental heart attack, all kinds of molecules were exposed to the immune system and the circulation such that an inflammatory response could be initiated, but having a Treg response to just one quieted the whole thing down. Activation of a Treg lymphocyte is restricted to its specific antigenic determinant. But immune suppression, immune modulation carried out by Treg cells is not specific to a given antigen. So the more Treg cells that we can get into the heart, the circulation or artery wall, the greater will be overall immune modulation, because these Treg cells responding to one antigenic determinant will release interleukin-10, transforming growth factor beta, that will suppress all the other pathologically activated cells in the circulation, in the heart, in the artery wall, or in the organ undergoing autonomic attack. And with fish oil and vitamin D, we change the entire milieu of your body to favor a Treg response. Now, you won't be surprised to learn that the closer you live to the equator, the lower is the incidence of cardiovascular disease. Also, the lower is the incidence of autoimmune disease and cancer. The more vitamin D your body receives, the better will your immune system function. Maladaptive responses such as atherosclerosis and autoimmune disease such as multiple sclerosis will be attenuated. Proper function of the immune system, such as killing cancers, will be enhanced. Now, we're all worried about sunlight because of skin cancer. I acknowledge that problem, and it's not really the sun that's causing skin cancer, it's free radical stress in the skin from sunburn. But skin cancers are fairly easy to deal with. But the closer you live to the equator, the lower will be your risk of the more threatening internal malignancies, because the more sun you get, the more vitamin D you have, the better maintained will be immune function. So the solution here is not to get a sunburn, but protect yourself from the sun in a reasonable fashion, and then take a lot of vitamin D, you'll get the same benefits. And it's been shown that in a meta-analysis of a number of studies that have been published, vitamin D supplementation decreases your cardiovascular mortality and all-cause mortality risk by about 10%. In a study of women who lived in Nebraska who did not have cancer at baseline, they were randomized to receive placebo or a small dose of vitamin D. I think it was 1 to 2,000 IUs a day. And over a 6-year follow-up period, new onset cancer rate was decreased by 50%. Pretty powerful protection, and protection that makes sense to us. Now, let's look at the effect of vitamin D on the activity of antigen-presenting cells, dendritic cells, and on Treg activity. We're going to look at 6-week-old ApoE knockout female mice. Now, these mice will develop atherosclerosis on a regular diet or on an atherogenic diet. They all receive a standard diet. They're going to have plaque formation, as you would expect. You add to their diet low-dose calcitriol or moderate-dose calcitriol, that's the active form of vitamin D, and a third group gets vehicle, that's the control group, the placebo group. You're going to sacrifice animals at 18 weeks and examine T cells and dendritic cells from the gut wall and from the spleen. So the gut wall will reflect local activity of vitamin D that is taken orally. The spleen will reflect systemic activity of the vitamin D absorbed from the gut, and we're going to look at arterial wall plaque. Low-dose vitamin D didn't do much to the vitamin D levels, but it rose significantly with moderate-dose vitamin D supplementation of the diet. Now, here we have the aortic root again. Plaque area was not significantly attenuated by low-dose vitamin D, but by high-dose vitamin D, it was significantly attenuated. They had less plaque formation, as you would expect, because they have a greater Treg response, more interleukin-10, more TGF-beta. Now, if we look at the number of Treg cells, those are FOXP3 cells in the bowel wall, we can see that oral vitamin D increases the frequency of Treg cells in the bowel wall. If we look at messenger RNA levels reflecting transcription of DNA in the gut and in the spleen, so we have local and systemic responses to vitamin D, we have greater levels of FOXP3, so we have more Treg cells. We have greater levels of CTLA4, that's the protein bound to the cell wall of Treg cells that suppress other immune cells, and we have greater levels of the anti-inflammatory cytokines released by Treg cells, IL-10 and TGF-beta. If we take T cells from the spleen and we stimulate them by reacting them with a monoclonal antibody to their CD3 receptor, their CD28 receptor, that should stimulate all T cells to proliferate. We see that the proliferative response is attenuated by vitamin D. You can still respond appropriately, but it is attenuated. So you are going to tamp down the immune response so it doesn't go out of control. If we look at levels of mature dendritic cells, those are the ones that are going to be expressing co-stimulatory molecules, so the T cells that they react with go Th1 or Th17. Those levels are attenuated in the gut and in the spleen by vitamin D. If we look at levels of IL-12, that is made by dendritic cells to encourage a T cell to go inflammatory, they are attenuated by vitamin D. IL-10 released by dendritic cells to promote a Treg response is increased. The dendritic cells will have on their cell surface molecules that can attract different types of T cells. These CCL17 and CCL22 attract Treg cells that are upregulated. So the vitamin D is affecting the characteristics not just of the cytokine milieu, not just of the T cells, but of the dendritic cells. It's arresting their development such that they will present antigen to T cells in a fashion to coerce them to take the Treg route. If we're looking at plaque now, we're giving the vitamin D orally. We've demonstrated an effect in the gut, in the spleen. Now we're going to look in the artery wall of these animals that are predisposed to atherosclerosis. We see more Treg cells in the artery wall. We see fewer activated dendritic or antigenic presenting cells in the artery wall. We see fewer macrophages. We see fewer T cells. We see lower expression of the monocyte adhesion molecules, VCAM and ICAM, on the endothelial surface, so they're not pulling in monocytes to become macrophages. We see lower levels of monocyte chemotactic protein, so they're not encouraging the bone marrow to spit out more immune cells to cause the plaque to propagate and activate. If we look at levels of interferon gamma and IL-6 in the plaque, there's less. We see more TGF-beta and IL-10 in the plaque. We see less IL-12 in the plaque, and we see increased levels of the molecules expressed by dendritic cells to call in Treg cells. So we're creating a T regulatory milieu in the gut, in the circulation, in the spleen, and in the plaque. This is why vitamin D is so helpful in all autoimmune conditions, including atherosclerosis and heart failure. Again, the dendritic cells and the immune cells are both released from the bone marrow. They share a similar progenitor cell type, so they interact closely. As a matter of fact, dendritic cells may be able to function as T cells and vice versa. Vitamin D will affect the dendritic cells such that they predispose the immune response away from Th1 and Th17 and towards Treg, which is in our favor if we have atherosclerosis or heart disease or autoimmune disease, or if we don't want to have those. Now let's look at the effects of fish oil. Will it have the same beneficial effects on immune system balance as will vitamin D? When we talk about fish oils, we're talking about eicosanoids. This is a slide from a presentation that we give on fish oil. And in years past, we did not really understand that fish oil works as an immune modulator. We thought that fish oil supplementation was useful to shift the balance of prostaglandins and leukotrienes. Essential fatty acids are fatty acids the body cannot make. We get them from the diet. We don't use them for fuel or structure, but we convert them into locally acting hormones called prostaglandins and leukotrienes. Whether we have anti-inflammatory prostaglandins or inflammatory prostaglandins and leukotrienes is really a function of the quality of the fish oils that we take in. Mother Nature designed us to take in relatively low levels of saturated fat and lots of unsaturated fat, thus we will have balanced prostaglandins. But if you look at the change in the American diet over the last 200 years, total fat intake has increased, and the quality of the fat that we take in has also changed. We are taking in saturated fats and the man-made trans fats. The unsaturated fats that we're taking in less omega-3 fish oil and more omega-6 oils from cereal grains. And that's not working in our benefit. We're supposed to have a balance. We're supposed to have some vegetable oils, some meat and dairy that gives us arachidonic acid, and lots of fish oil, so we have a balance. However, we don't follow a balanced diet. We don't take in much fish oil. We don't take in proper amounts of unsaturated vegetable oils. We're getting a lot of meat and dairy. We're out of balance. We're making a lot of inflammatory prostaglandins, such as arachidonic acid. Now, what we want to do is cut back on red meat and dairy, take in beneficial essential fatty acids from fish oil or from gamma-linolenic acid from borage oil or evening primrose oil, and get back into balance. Again, the quality of our prostaglandins is a reflection of the quality of our dietary fatty acid intake. However, the enzyme systems that convert the dietary fatty acids into the prostaglandins and leukotrienes can be influenced by our therapy. In a preceding section, we talked about how if we block the activity of angiotensin II in the vessel wall, we will blunt the conversion of arachidonic acid into the matrix metalloproteinases that degrade the fibrous plaque and cause plaque rupture. Let's see if fish oils will have a similar effect on prostaglandin physiology. Now, we know that fish oils have great benefit in cardiovascular disease, and this slide is about 7 years old before I knew a thing about the immune system. And we've learned over the years that fish oil supplementation is of value in essentially every known cardiovascular condition. And when I made up this slide focusing on heart disease, I came across articles showing benefits of fish oil in other conditions such as lupus or asthma. And I thought this had to do with prostaglandins, and it did, but it also has to do with the immune modulatory effects of fish oil. So we have a 2-hour presentation on the cardiovascular benefits of fish oil. Here we're going to talk about the immune modulatory effects. Again, let's look at this sequence of events. An antigen capture cell, a dendritic cell, recognizes and engulfs an invader. It hightails it to some immune tissues, and in this process, it converts itself from an antigen capture cell to an antigen presenting cell, now armed with co-stimulatory molecules. It presents the antigen determined and the co-stimulatory molecule to the naive T cell. Now, a T cell will activate as a Th1 if it is exposed to its cognate antigen and if the antigen presenting cell is armed with co-stimulatory molecules. And if it's going to go Th1, it will elaborate interleukin 12, interleukin 6, TNF-alpha, interferon gamma, drive forward other immune cells. It utilizes some membrane-bound molecules, CD28 and CD40 ligand, to contact activate other immune cells. So the molecules it secretes will affect the other immune cells in the milieu, but there's also direct cell-to-cell interactions with one molecule binding or ligating another receptor. Now, immune system downregulation must follow immune system activation to prevent excessive tissue damage from inflammatory cytokine activity and to avoid autoimmune conversion. If we have pneumococcal pneumonia, we want a brisk Th1, Th17 immune response to kill the bugs, and then we want to quiet down, and when we no longer need the immune response, we want to quiet it down. Now, Treg cells fulfill this function. Treg cells display CTLA4, that's called cytotoxic lymphocyte antigen 4. We're going to call it a suppressive molecule that can interact with a receptor on all the other immune cells to quiet things down. The T helper cells that are active express CD28. The Treg cells express CTLA4 to quiet things down. Now, all Th1 and Th17 cells can express CD28, which is stimulatory, and CTLA4, which is suppressor. You want high levels of CD28 early in response to stimulation to go kill the bugs, and then you see CTLA4 2 to 3 days later. B17 on the immune cells receiving these signals binds 20 times more avidly to the suppressor CTLA4 than to the stimulator CD28. Okay, so our immune system is more sensitive to suppression than activation. CTLA4 ligation shuts down T cell proliferation and cytokine response. We want to go kill the bugs, and then it's very important to quiet down, and that involves Treg function, the soluble cytokines IL-10 and TGF-beta, and the membrane-bound molecule CTLA4. Balance between CTLA4 and B7 protects against inappropriate or prolonged inflammation. Now, let's look at the effect of EPA, that's the essential fatty acid found in fish oil along with DHA, on CTLA4 expression. We're going to take healthy mice, assign them to 1 of 3 diets. The control group gets 5% corn oil, which is saturated fat. One group gets 4% corn oil and 1% DHA. The other group gets 4% corn oil, 1% EPA. 2 weeks later, we're going to study T lymphocyte function, T cell proliferation in response to non-specific stimulation of CD3 and CD28, which is supposed to cause the T cells to proliferate, and cell membrane expression of CD28, which is active, versus CTLA4, which is suppressive. Now, the T cells at rest do not proliferate, but if you expose them to a monoclonal antibody that's supposed to make them proliferate, they will all increase their proliferation rate. And it's much greater if the animals receive saturated fat corn oil. It's attenuated with both DHA and EPA. If we look at CD28 surface expression, which is appropriately upregulated in a physiologic immune response, it's the same in all 3 groups. Fish oil will not inhibit appropriate immune activity or T cell proliferation. But if we look at CTLA expression, which should quiet things down, that's physiologic, it is delayed with the corn oil group. DHA doesn't have much effect, but it is accelerated with fish oil. This is one of the means by which fish oil quiets down an uncontrolled immune response by accelerating the expression of the suppressive CTLA4. Let's look at the effect of EPA and DHA on monocyte gene expression, the proteins that are going to be transcribed from the genetic code of the monocyte. We're going to look at 111 healthy non-obese subjects. We'll do baseline blood studies, randomize them to receive 6 gel caps to provide sunflower oil, low-dose fish oil, high-dose fish oil, repeat the lab studies at 26 weeks. Now, with essential fatty acid, particularly fish oil supplementation, we expect to see a decrease in triglycerides. And with low-dose fish oil, there's a 5-point decrease. With high-dose fish oil, 28-point decrease. We would expect that. The decrease in fatty acids is greater with high-dose fish oil than low-dose fish oil. Fish oil blunts insulin insensitivity. When we're insulin insensitive, we go into storage mode, make triglycerides, release fatty acids in the blood. We don't like that. Fish oil supplementation via an anti-inflammatory pathway improves insulin sensitivity, triglycerides fall, free fatty acids fall. The percentage of DHA and EPA in the total fatty acids rises. It rises a little with low dose, it rises a lot more with high dose fish oil. So we're replacing other fats with the fish oil, EPA, and DHA. We would expect that. Now, if you look at the gene products being produced from the DNA, the same DNA of the mononuclear cells, multiple proteins, their synthetic rate is affected with fish oil supplementation. Over 1,000 genes are affected, and the gene families downregulated are those for adipogenesis, so you make fewer fat cells. Inflammation, endothelial adhesion molecules, the scavenger receptor on macrophages, eicosanoid metabolism, that's fatty acid metabolism, goes anti-inflammatory. Those toll-like receptors that upregulate macrophage function, they are decreased with fish oil. Oxidative stress, less nuclear factor kappa beta. So fish oil has an important effect on immune function by changing the way you're going to read your genetic code towards a non-inflammatory pathways. Let's look at the effect of EPA and DHA on monocyte Th1 cytokine production. We would expect to see lower levels of Th1 cytokines. 16 young men, baseline measurements, supplement them over successive 4-week periods with EPA and DHA at 300 milligrams a day, 1,000 milligrams a day, 3,000 milligrams a day. Repeat the baseline measurements following 4 weeks at each dose. You can see with increasing doses of fish oil, you will see an increasing percentage of cell membrane fatty acids composed of EPA and DHA. We would expect that. If we look at the amount of interleukin-6 produced by the monocytes, blue is at rest and red is in response to stimulation. You can see that on the control diet, at rest and with stimulation, you're making a lot more IL-6. In the young men treated with fish oil, you make less IL-6 at rest and less with stimulation. That is desirable if we want to prevent cardiovascular disease and autoimmune disease. The same situation with tumor necrosis factor alpha. It is quieting down the Th1, Th17 immune bias that Americans have. What effect will fish oil have on another autoimmune disease, such as exercise-induced asthma? One of the neat things about nutritional medicine is if a supplement quiets down abnormal physiology or autoimmunity in one organ, we can anticipate it will do the same in every other organ of the body without a downside. Let's see if fish oil will help with asthma. 20 ranked college athletes in the UK. 10 had exercise-induced asthma. With physical activity, they would experience shortness of breath. It was worse in the colder months. FEV1, how much air you can exhale in 1 second, an objective marker of asthma, dropped with exercise. And 10 other athletes did not have exercise-induced asthma. You'll do a baseline assessment on the usual diet, then randomize them to receive 18 capsules a day over 3 weeks to provide high-dose fish oil or olive oil placebo. This was not extra virgin olive oil that is beneficial as an antioxidant. This is grocery store olive oil that is oxidized that really doesn't have much of a physiologic effect. And you're going to cross them over to the opposite treatment after a 2-week washout period. Double-blind protocols followed. Interleukin-1 beta and tumor necrosis factor alpha, Th1, Th17 cytokines. Here we see on the normal diet, interleukin-1 beta pre-exercise and following exercise it rises. Here we have the olive oil group on their control diet, the olive oil diet, and here's after exercise, there's no real change. So the olive oil placebo is not blunting the Th1, Th17 response to exercise. However, in the fish oil group, you can see here in blue is the control diet. On fish oil, they're releasing less interleukin-1 beta at rest and much less interleukin-1 beta following exercise. The same thing with tumor necrosis factor alpha. So the driving force underlying their exercise-induced asthma, the cytokines, is attenuated with fish oil. Plasma leukotriene B4 and urine leukotriene B4, markers of inflammatory leukotriene products of essential fatty acids, were likewise attenuated in the fish oil group, not in the olive oil placebo group. And if we look at FEV1, it decreases with exercise on the control diet, on the olive oil diet. It doesn't decrease much at all on the fish oil diet. And how often they need the rescue inhalers? 29 puffs per week on their usual diet, 28 puffs per week with the oxidized olive oil, only 19 puffs per week for fish oil. So fish oil supplementation quiets down the autoimmune condition, exercise-induced asthma. Essential fatty acids in lupus. This slide is 7 years old before I understood anything about T cells or the immune response. And this comes from our talk on essential fatty acids. In heart disease, and I presented some studies showing that essential fatty acids are helpful in some non-cardiovascular diseases. Now, lupus, systemic lupus erythematosus, is a chronic disorder characterized by arthritis, rash, vasculitis, blood vessel inflammation, inflammatory damage to the nervous system, the kidneys, the heart, and the lung. It is a disease of unknown cause with an 8-to-1 female-to-male ratio. That's because testosterone, as we'll talk about soon, suppresses Th1, Th17, whereas estradiol does not. Now, lupus is autoimmune in nature. T cells are reacting to self-membrane proteins as non-self. They're making a mistake, just as they make a mistake when they respond to oxidized LDL in the artery wall and release inflammatory cytokines. B cells produce abnormal self-reacting antibodies. The antibodies deposit in joints and other target organs. Prostaglandins mediate much of the tissue damage. Thus, fish oils affect prostaglandin metabolism. We felt that fish oil might help in lupus by altering prostaglandin metabolism. And it's considered to be an incurable disease. The treatment involves reducing inflammation and symptoms with aspirin, prednisone, and nonsteroidals, or suppressing the out-of-control immune system with immunosuppressive and cytotoxic therapies. Chemotherapy to quiet down, to kill off all immune cells, good and bad. That will improve your symptoms, but of course, by suppressing the entire immune system, it will increase your risk for other health conditions. Now, levels of several cytokines are abnormally elevated. Th1, Th17 cytokines, interleukin-1 beta, interferon gamma, TNF-alpha, and interleukin-2, which is released by activated immune cells to stimulate their own proliferation, the proliferation of other immune cells in the milieu. All four are pro-inflammatory, all attract other white cells to the site of inflammation, stimulate these white cells to make inflammatory prostaglandins, free radicals, inflammatory cytokines, and leukotrienes. Interleukin-2 stimulates further autoreactive T cell and antibody production. We get a vicious cycle: symptoms, organ system dysfunction, debility, and death. Now, much of the tissue damage is actually carried out by free radicals, inflammatory prostaglandins, leukotrienes, and cytokines. Omega-3 essential fatty acids will block production of inflammatory prostaglandins and leukotrienes, promote production of anti-inflammatory prostaglandins, inhibit production of inflammatory cytokines. Could omega-3 deficiency be a cause of lupus? Will omega-3 supplementation be of value in patients with lupus? Let's take a look. 12 patients with newly diagnosed lupus, and as a control group, you draw blood from healthy volunteers. You do the baseline measurements and then treat them all with fish oils to provide just under 1,000 milligrams of EPA and 864 milligrams of DHA. That would be about 6 to 8 gel caps of fish oil. Patients who had kidney or heart involvement received steroids during months 1 and 2, and then the steroid therapy was stopped. They all received fish oil, and you're going to reassess at 6 months. Let's look at the percent distribution of plasma fatty acids. In the healthy controls, the patient with the lupus, there's very low levels of EPA. With supplementation with EPA, it rises. The lupus patients had lower levels of DHA than did the controls. With supplementation, it rises. They have higher levels of the inflammatory fatty acid, arachidonic, than did controls, and you supplement with fish oils, that falls. When you supplement with one type of essential fatty acid, levels of the other ones, the bad guys, tend to fall in lockstep. Now, let's look at levels of lipid peroxides. That's like malondialdehyde, a marker of free radical stress. It was greater in the lupus patients than in the healthy controls. With fish oil supplementation, it falls. Superoxide dismutase is a key selenium-dependent antioxidant enzyme system. 1/5 of the protein in your body is superoxide dismutase because our antioxidant defense are so important. These levels were low in the lupus patients because of their unremitting free radical stress. You give them fish oil, you quiet down inflammation, levels of superoxide dismutase rise. Nitric oxide is elaborated by endothelial cells to provide the Teflon coating so the blood vessels dilate and resist platelet clotting. It was lower in the lupus patients. Given fish oil, it rises. Glutathione is our key intracellular antioxidant. It was depleted in the lupus patients. You give them fish oil, it increases. So, with omega-3 supplementation in patients with lupus, arachidonic acid levels fall, anti-inflammatory omega-3 and omega-6 fatty acid levels rise, antioxidant enzyme systems are elevated, lipid peroxides, markers of free radical stress, fall, nitric oxide and glutathione levels rise. Well, biochemically they got better. How did they do clinically? They all went to remission. They got these people early, intervened with an immune modulator, fish oil, and they got better, as you would expect. Let's look at the effect of essential fatty acids on Th1 versus Th2 in cytokine production. Study of healthy mice assigned them to 1 of 4 diets. A low-fat diet, standard chow containing corn oil, a high saturated fat diet, chow with coconut oil, which is saturated, high omega-6 chow with safflower oil, high omega-3 chow with fish oil. 6 weeks later, we're going to study T lymphocyte structure and function. And if we supplement you with safflower oil, we see some gamma-linolenic acid, which is a good omega-6. If we supplement you with fish oil, we see a lot of EPA, which is the beneficial omega-3. Now, thymidine incorporation, that's lymphocyte proliferation rate, following stimulation with Con-A, that is a nonspecific mitogen. It causes T cells to proliferate in general. In the animals fed saturated fats, they proliferate much more avidly. Why? Because we're stimulating that toll-like 4 receptor, so we're kind of putting a hair trigger on the immune response, so they're going to proliferate more aggressively. Safflower oil provides some protection, fish oil the most protection. We look at IL-2 production. It is greatest in the saturated fat diet. IL-2 is released by immune cells to cause them to proliferate and to cause other cells in the immune to proliferate. That is increased with saturated fat. Now remember, IL-2, the drugs and drug-eluting stents block IL-2. That's pretty darn important, isn't it? With saturated fat intake, our whole body becomes an IL-2 producing organ, so to speak. There's some protection with the omega-6 and with the fish oil. So there's a reduction there. Interferon gamma, which is Th1, Th17, is elaborated at a higher concentration with saturated fat. There's some protection with the beneficial omega-6, more protection with fish oil. Interleukin-4, which is a Th2 cytokine, was not affected. If we look at the ratio of interferon gamma to interleukin-4, it was high with saturated fats. There was some protection with the safflower oil, which is the beneficial omega-6. The greatest protection was with fish oil. So saturated fat cranks up lymphocyte sensitivity to any stimuli, and it skews you towards Th1. Fish oil quiets things down, and it particularly quiets down Th1, but it doesn't quiet down Th2 as much, so the balance goes more towards Th2, and as we'll see later, more towards Treg. If we look at the messenger RNA following stimulation, at 4 hours and 8 hours, it will rise with chow. It's greater at rest, and it rises over time more with saturated fat. The omega-6 doesn't have much effect. Fish oil has the greatest effect. If we look at messenger RNA for interferon gamma, we see the same thing. So fish oil is a protective. It promotes a regulatory response. It promotes a Th2 response, and it downregulates the Th1 response. Another study, healthy mice assigned to one of two diets, standard chow with corn oil, chow with 1% corn oil and 4% fish oil. 2 weeks later, you study T cell proliferation. If the milieu was adjusted to promote Th2 differentiation, fish oil had no effect. If the milieu was such that the T cells tended to go Th1, proliferation was suppressed. Fish oil doesn't seem to dampen a Th2 response, but it does dampen a Th1 response. As autoimmune disease in humans, particularly atherosclerosis, heart failure, or rheumatoid arthritis, or asthma, are Th1 conditions, fish oil supplementation tends to bring you back into balance. We want to stay in balance, because T cell activation pathway determines cytokine milieu. Cytokine milieu determines T cell activation pathway and whether you do or do not develop autoimmune disease. I couldn't find a paper on fish oil in adult heart failure. In our 2-hour presentation on fish oil, I present a number of studies showing benefits of fish oil and atherosclerosis, but I did find a paper on fish oil in pediatric cardiomyopathy. 28 kids with cardiomyopathy, and they did not have coronary disease or valve disease. They had a cardiomyopathy, an autoimmune condition. Mean age is 10 years, all on standard therapy. They'd been under standard therapy for 5 years. You do baseline measurements, randomize them to receive over 6 months cod liver oil, giving them about 1,500 milligrams of EPA and DHA or no supplement, and you had healthy kids for the comparison group. Their hearts were dilated. The left ventricular diastolic dimension, the dimension of the heart at rest, was obviously greater in the kids with cardiomyopathy than in the healthy kids. Over 6 months with placebo group, there was minimal change. There was some decrease in chamber size with fish oil. Ejection fractions were lower. It was 60 68% in healthy kids, didn't change over time. In those without fish oil, it didn't change significantly, and it rose from 48% to 56% with fish oil. So pump function improved. The heart was smaller, giving these kids with what we presume to be an autoimmune heart disease, fish oil. So if we ever do a biopsy of your heart, this is something that we want to see. There ought to be something fishy going on in your heart. So vitamin E and fish oil promote a Treg response. They quiet down the deleterious Th1 and Th17, thus they're beneficial in atherosclerosis, heart failure, and all autoimmune conditions. Let's move on now and discuss altering the cytokine milieu with testosterone. We'll continue our discussion of milieu therapy with this section on testosterone. Now, when we talk about risk factors, the causes of cardiovascular disease that we're aware of, we've learned that testosterone deficiency underlies or aggravates many of our standard cardiovascular risk factors, and we know that testosterone deficiency is an independent risk factor or cause of cardiovascular disease. We have a separate 2-DVD presentation on the benefits of testosterone in heart disease. Here I'm going to be focusing on the immune benefits of testosterone in cardiovascular disease. There are multiple links between low testosterone and cardiovascular disease. We touched upon lipoprotein lipase, insulin sensitivity, lipids and lipoprotein A, but we also know that low testosterone in men is associated with persistent inflammation and high Th1, Th17, and low Treg immune dysregulation. In response to testosterone supplementation, we keep nuclear factor kappa beta from translocating to the nucleus. So we see lower levels of IL-6, interleukin-1 beta, and CRP. We see lower levels of the white cell endothelial interaction molecules, VCAM and ICAM. We see lower levels of the degradative enzymes, matrix metalloproteinases. We see higher levels of the Treg cytokine, IL-10. So thus we would expect that testosterone would be of value in atherosclerosis and heart failure. Indeed it is. Testosterone will affect the cytokine milieu. Cytokines are the driving force behind cardiovascular disease. Let's see what effect testosterone has. And we also know that the matrix metalloproteinases degrade the fibrous cap. If we can keep those levels down, we're going to promote stabilization of the plaque, lower your risk of unstable angina or heart attack. Well, let's start by a discussion of testosterone in another Th1, Th17 disease, rheumatoid arthritis. 36 men with long-standing rheumatoid arthritis and 70 age-matched healthy controls. 24 of the 36 men with RA were receiving prednisone, a corticosteroid. 12 were not on prednisone. Testosterone levels in the patients with RA not on prednisone were slightly lower than those of age-matched healthy controls. Those on prednisone were quite low. Prednisone is a degradative catabolic influence, and it's associated with lower testosterone levels. Luteinizing hormone is made in the pituitary gland upon instruction from the hypothalamus to tell the testicles to make testosterone. In the men with rheumatoid arthritis not on prednisone, they had lower levels of testosterone and their brains responding by making luteinizing hormone. Those on prednisone did not. So the prednisone was affecting brain function as well as testicular function. So rheumatoid arthritis is associated with low testosterone. Prednisone corticosteroid therapy lowers testosterone further. Now, a study of therapy. 7 men with rheumatoid arthritis, 8 controls that have osteoarthritis, age-related, non-autoimmune arthritis, all on non-steroidal therapy like Motrin, not on prednisone. We're going to do baseline measurements, treat the RA patients with testosterone, oral testosterone 3 times a day, reevaluate at 6 months. Testosterone levels were lower in the patients at baseline. They normalized with supplementation. Now, here we're going to look at T-cell subsets. This is a very old paper before they knew about Th1, Th17, Treg, and Th2. Back then, you looked at CD4+, which is all of the T helper cells, versus CD8 cells. CD8 cells, we don't talk about them much in a discussion of atherosclerosis and heart failure. Their job is to kill cells that are infected with viruses or cells that have gone cancerous. In autoimmune conditions like RA, you tend to see a high ratio of CD4 plus to CD8 plus. And this is what you saw. There were lower levels of CD8 plus cells in the RA patients. In response to testosterone, the levels increased. So the ratio of CD4 plus to CD8 plus was high in the RA patients at baseline. It improved with testosterone. The erythrocyte sedimentation rate, that's kind of like C-reactive protein. It's an old-fashioned marker of inflammation. It fell with testosterone. Rheumatoid factor is a marker of the activation level of your RA. It also fell. The number of tender joints fell, and the requirement for motion light agents fell. So testosterone is helpful in rheumatoid arthritis. A Th1, Th17 high, low Treg disease, and so thus you would expect that it would work in all autoimmune conditions such as atherosclerosis and heart failure. Let's look at the interaction between testosterone, the extent of coronary disease, and cytokine levels. 69 men with angiographically proven coronary disease. 20 had one vessel involvement, 18 had two vessel involvement, 31 had three vessel disease. 17 were hypogonadal, they had low testosterone levels. 22 were borderline, 30 had normal testosterone levels. In this group of men with known cardiovascular disease, some with one, some with two, some with three vessel disease, some with low, some with normal testosterone levels, let's look at levels of inflammatory cytokines, TNF, interleukin-1 beta, interleukin-6, and the anti-inflammatory Treg cytokine IL-10. We're going to look for correlations and relationships. Now, interleukin-1 beta, an inflammatory Th1, Th17 cytokine, was greater in lockstep with the extent of cardiovascular disease. As you go from 1, 2, 3 vessel involvement, higher levels of interleukin-1 beta. The subsequent 3 vessel disease had higher levels of interleukin-6 than those with 1 or 2 vessel disease. The interleukin-10 was also increased in the patients with more extensive cardiovascular disease. Now, at first, that seems kind of paradoxical, doesn't it? IL-10 is a suppressive cytokine. But as we learned in our discussion of the frequency of Treg cells in humans with various degrees of atherosclerotic plaquing, the more complicated or advanced the plaque, Mother Nature will begin to call in a Treg response. So we have inflammation going on in the patients with extensive cardiovascular disease. You do pull in a Treg response, but it doesn't work so well. Therefore, the disease progresses. These patients all had stable atherosclerosis. And so the interleukin-10 being released by the Treg cells is kind of neutralizing the interleukin-1 beta, the TNF, and the interleukin-6. In patients with unstable disease, where Treg suppression has been lost, we would expect to see lower levels of IL-10. Now, here we see that IL-1 beta rises in proportion to the extent of the cardiovascular disease. Here we're going to see that IL-1 beta rises in relation to the testosterone level. Individuals with normal testosterone levels have lower levels of interleukin-1 beta. Those with slightly low levels have an intermediate level. Those that are hypogonadal with low testosterone, they have higher interleukin-1 beta. So the lower your testosterone, the more Th1, Th17 cytokine production, the more extensive will be your cardiovascular disease. That makes sense to us. Now, so if low testosterone is associated with high levels of inflammation, what's going to happen if we treat you with testosterone? Will Th1, Th17 cytokine levels fall? Let's look at the effect of testosterone on C-reactive protein in elderly men. C-reactive protein is made by the liver in response to Th1 cytokines such as IL-6. And it's the lab marker that we are utilizing to assess systemic whole body inflammation. 60 middle-aged elderly men presenting to a clinic with urologic complaints. All are hypogonadal with low testosterone levels. You'll do a baseline CRP, and then you'll assess the severity of their symptoms with the aging male symptom scale. You're going to treat them all with testosterone, reevaluate 1 year later. Their symptoms decreased with testosterone. They felt better. And look at their C-reactive protein. 3.5 at baseline, 2.6 at 6 months, 2.2 at 12 months. Th1, Th17 inflammation falls, CRP falls. They felt that their symptoms were related to the degree of inflammation. The higher the CRP, the worse they felt. You lower inflammation, you're lowering CRP, and you're making people feel better. We don't feel well when we're inflamed. When you lose weight, inflammation falls, you feel better. When we replace testosterone, inflammation falls, and you feel better. Another study of testosterone inflammation, 25 men attending an outpatient andrology clinic. And 20 of the men had coronary disease, 10 were hypertensive, 2 had heart failure, 9 also had diabetes. So these are typical elderly patients with cardiovascular disease and low testosterone. Do baseline lab studies, randomize them to receive on days 1, 14, and 28 intramuscular testosterone or placebo, repeat the lab studies on day 30. And with placebo therapy, not much happens with the testosterone level, nor would we expect it. Of course, it rises significantly with testosterone replacement therapy. Tumor necrosis factor alpha rose in the placebo group. It fell with testosterone, just as CRP did in the preceding study. Interleukin-1 beta falls. Interleukin-10, the good guy from a Treg cytokine, rises, just as it does with vitamin D, just as it does with fish oil. In this study, we're going to take these New Zealand white rabbits, randomize them to standard diet, sham operation, no hormonal interventions. Another group are put on a high cholesterol diet, a sham operation, no hormonal intervention. Another group of rabbits are put on the bad diet, they're castrated with no hormonal intervention. Another group, bad diet, they're castrated and they receive testosterone replacement therapy. Sacrifice them all at 12 weeks and evaluate aortic wall, plaque, and pertinent lab studies. Testosterone levels rise in response to a high cholesterol diet. If we eat a lot of red meat with cholesterol, we will make more testosterone because testosterone is made in the body from cholesterol. The levels don't rise a lot, but they rose in this study where the animals were also fed an abnormal diet. Now, if you castrate the animals, you can see that testosterone levels are quite low. If you remove the testicles from an animal, their adrenal gland will still make some testosterone. That's why the levels weren't zero. The animals on the high cholesterol diet that were castrated and then received testosterone replacement therapy, they got the job done with a physiologic testosterone level. If you look at the percentage of the lumen, the flow area involved with plaque, with a standard diet and no surgery or a sham surgery, there's no plaque. On the high cholesterol diet and sham surgery, there's plaque. High cholesterol diet and castration, so you have testosterone deficiency and a high cholesterol diet, you have a lot of plaque. Castration, bad diet, and testosterone replacement therapy, it is attenuated. So the low testosterone and a high cholesterol diet interact to produce more plaque. If you replace the testosterone, you can regress the disease correspondingly. Thickness of the fibrous cap. We want a thick fibrous cap full of collagen to keep the plaque from becoming vulnerable. In the animals on a high cholesterol diet who were castrated that had low testosterone, the fibrous cap is thinner. If you give them back the testosterone, it thickens, making the plaque less likely to rupture or ulcerate. You want lots of collagen in your plaque, and without testosterone to restrain the immune system, matrix metalloproteinases are elaborated and it erodes the plaque. You give the animals back testosterone, the fibrous cap thickens again. Inflammatory cytokine levels. If we look at IL-6, soluble, ICAM, and matrix metalloproteinases, the ICAM is elaborated by the endothelial cells to pull in white cells. We don't like those. Those are all bad guys. This is standard diet and sham surgery. High cholesterol diet, they're all higher. High cholesterol diet and castration to create a low testosterone situation. Again, there's this unfavorable interaction between a high cholesterol diet and low testosterone, more inflammatory cytokines. You replace the testosterone, you can replace that. Looking at it differently, here's IL-6, here's ICAM, here's MMP. High cholesterol diet will increase it. High cholesterol diet, low testosterone makes it even worse. Testosterone and nuclear factor kappa beta translocation. Because testosterone is lowering all these inflammatory mediators, how does it do it? We would predict that it would keep tumor necrosis factor alpha out of the nucleus, similar to how ECP would work. Here we're going to take healthy endothelial cells from human umbilical veins of newborns, put them in tissue culture, and we're going to look at nuclear factor kappa beta nuclear translocation and two indicator molecules. Tissue factor pathway inhibitor is a good guy, and we're going to look at this anti-inflammatory protein and its messenger RNA. Before and after you incubate these cells with testosterone, tumor necrosis factor alpha, or both. Now, tissue factor pathway inhibitor, a good guy, an anti-inflammatory molecule, is increased with testosterone. It is decreased with tumor necrosis factor alpha, and that deleterious TNF effect is attenuated with testosterone if you go with the protein and the messenger RNA. So the TNF-alpha is changing the way we read DNA in an unfavorable fashion. Testosterone changes the way we read our DNA in a favorable fashion. Testosterone will attenuate many of the negative consequences of Th1 cytokines such as TNF. If we look at nuclear factor kappa beta binding within the nucleus, this is attenuated by testosterone. This is why testosterone has a broad spectrum anti-Th1, anti-Th17 effect. Tumor necrosis factor alpha increases it. The deleterious effect of TNF-alpha is attenuated by testosterone. Thus, testosterone would be therapeutic in diseases associated with active inflammation such as rheumatoid arthritis, atherosclerosis, and heart failure. Look at the effect of inflammation, TNF, on testicular function. 80 male Wistar rats, baseline measurements, and treat them with an intravenous infusion of TPN, total parental nutrition. That is the nutritional source you would receive if you were in an intensive care unit. This was a model of post-surgical ICU situation. So one group gets total parental nutrition, another group gets TPN and a moderate level of TNF, a third group gets TPN and a high level of TNF to kind of reflect a post-operative surgical sepsis situation. Sacrifice and evaluate 1/3 of each group at 1, 3, and 6 days. And what you find is the animals exposed to the inflammatory mediator TNF have lower levels of testosterone at day 1, day 3, and day 6. Testicular weight is decreased with inflammation. So testicles will shrink, they will become dysfunctional. Inflammation lowers testicular function. Belly fat in men is inflammatory. So if you have belly fat and a lot of Th1 cytokine activity, that will compromise testicular function. That is why the heavier you are with belly fat, the lower will be your testosterone levels. And the lower the testosterone, the greater will be your risk for heart disease, atherosclerosis, and heart failure, because you will lose the modulating effect of testosterone. Let's look at testosterone and the IL-10 to IL-12 balance. IL-10 is regulatory. It quiets things down. It's a Treg cytokine. IL-12 is a Th1, Th17 cytokine. We're going to look at T lymphocytes recovered from the spleens of male and female mice. Stimulate the T cells to proliferate with a nonspecific mitogen and measure cytokine production. Female mice make less IL-4 than do male mice. IL-4 is Th2. They make less IL-10, which is Treg. They make a lot more IL-12, which promotes Th1, the same amount of interferon gamma. So women who have high estradiol, less testosterone, are more likely, they're sort of skewed towards Th1 and away from Th2 and Treg, which is why women have more autoimmune disease than do men. So what they did, they took these female mice and treated them with testosterone. They gave them dihydrotestosterone, same thing as testosterone therapy. And they repeated the study, and the female mice that received testosterone shift to a more male cytokine pattern with more IL-10, more Treg, less IL-12, less Th1. Testosterone upregulates IL-10, Treg, downregulates IL-12, which is Th1. Testosterone inhibits Th1, promotes Treg immune responses. Thus, we understand that testosterone protects against autoimmune disease and atherosclerosis and heart failure. And testosterone keeps nuclear factor kappa beta out of the nucleus. Therefore, it's going to decrease production of the inflammatory cytokines, interleukin 6, interleukin 1 beta, and CRP, its marker. It increases the Treg cytokine IL-10. It lowers levels of adhesion molecules elaborated from the inflamed endothelial surface. It lowers levels of matrix metalloproteinases, thus protects our plaques from becoming vulnerable. If we're overweight, our visceral fat is releasing inflammatory mediators that cause the testicles to make less testosterone. With less testosterone, we tend to shunt fat from the periphery into the belly. We have more inflammation, less testosterone, more overweight, vicious cycles occur. With weight loss, testosterone levels will rise. With testosterone replacement, we tend to help shift fat out of the belly into the periphery, so there's a synergy there. There are all cardiovascular conditions, atherosclerosis and heart failure, have been shown to respond in a favorable effect to testosterone, and many non-cardiovascular conditions will respond to the anabolic benefits of testosterone. And here we discussed how testosterone shifts us from Th1, Th17 more towards Th2 and Treg. Milieu therapy, similar to that with vitamin D and fish oil. So all my patients are encouraged to take vitamin D and fish oil. All my male patients, we look for low testosterone, and it is very unusual for me to see a younger male patient with heart disease that does not have a low testosterone. And now let's move on to the next section, talking about pentoxifylline in the treatment of cardiovascular disease. Our last section discusses therapy with pentoxifylline and famotidine. Pentoxifylline is useful clinically because it carries out cytokine blockade. We've talked about tumor necrosis factor alpha. It is a Th1, Th17 inflammatory cytokine. If we look at the relationship between your TNF levels, functional class, and outcome in heart failure, as you go from normal to class 1 heart failure, that is nearly asymptomatic, to class 4 heart failure, that's symptoms at rest, the worse you are, the higher is your TNF-alpha level. That would make sense. If we look at outcome in relation to TNF levels or soluble TNF receptors, which is another marker of TNF activity, the greater your level of TNF, the worse is your outcome. So the worse your cardiovascular disease, the higher the TNF, the worse is your outcome. That makes sense because TNF is driving your heart failure. Now, let's look at some of the maladaptive responses of tumor necrosis factor alpha. Heart failure is associated with cytokine elaboration. Interleukin 6, CRP, and tumor necrosis factor alpha all transcribed under the influence of nuclear factor kappa beta. Why do we see high levels of inflammatory mediators in heart failure? Well, 5 to 10 years ago, we felt that the high levels of inflammation in heart failure were due only to translocation of bacteria across the edematous bowel wall. In heart failure, you retain fluid. You have peripheral edema. Your bowel wall will also swell up with fluids. And under these circumstances, bacteria can leave the gut and enter your circulation. And we will see levels of bacterial cell wall lipopolysaccharide in the system. That's why in heart failure, you often kind of look red and inflamed because bacteria are leaving the gut and entering the bowel wall. And that was the only interaction that we were aware of 5 to 10 years ago. Now we understand that heart failure, because it's associated with sympathetic activation, oxidative stress, high levels of angiotensin II, and inflammation, will lead to a Th1, Th17 low Treg response. Heart failure is associated with a maladaptive response to the immune system. What are the deleterious effects of high, non-physiologic levels of TNF-alpha? It has a negative inotropic effect. The heart contractility is decreased. It encourages the heart cells to commit suicide via a pathway called FAS-APO1. It increases the activity of matrix metalloproteinases. Those will degrade the fibrous plaque of an arterial lesion. It also causes the heart muscle cells to remodel and enlarge. It's going to degrade the cytoskeleton of the heart. It leads to endothelial dysfunction, so impaired blood flow to the heart muscle, and it tends to promote atherosclerosis. Now, if TNF is such a bad guy, let's knock it out. And we can do this with drug therapy. Etanercept, its trade name is Enbrel, blocks the TNF receptor. It just wipes it out completely. It is useful in the therapy of rheumatoid arthritis, another Th1, Th17 disease. It works great in rheumatoid arthritis. Well, let's try it in heart failure. Studies were done taking people with heart failure. They received placebo or etanercept to wipe out TNF, and the surprise finding was the treatment group did worse. Why is this occurring? Well, Mother Nature did not give us TNF to kill us. We need low levels of TNF for normal cell function. High levels of TNF are associated with suicide of the heart muscle cells. Low levels of TNF prevent the heart cells from committing apoptosis or cell suicide. You wipe it out completely, you get rid of the bad, you get rid of the good. So wiping out TNF completely is a bad idea. However, with agents such as pentoxifylline, we can modulate the transcription of TNF. We won't wipe it out completely, but we'll tamp it down when it is pathologically upregulated. So let's look at the effect of pentoxifylline that doesn't wipe out TNF, but which quiets it down in ischemic cardiomyopathy. 38 patients with heart failure, their ejection fractions were less than or equal to 40%. They all had extensive coronary disease. So their heart failure was due to prior heart attack and poor blood flow to the heart. They're all on treatment with ACE inhibitors, carvedilol, and most are receiving a diuretic or spironolactone, another neuroendocrine blockade agent. After 3 months of observation on their standard therapy, you randomize them to receive placebo 3 times a day or pentoxifylline 400 milligrams 3 times a day. Otherwise, similar care, their other drugs were not changed. You reevaluate at 6 months except for 5 patients who died, and 4 of the 5 who died were in the placebo group. A double-blind protocol was followed. CRP rose over 6 months with placebo therapy. It fell with pentoxifylline. TNF-alpha rose with placebo therapy. It fell with pentoxifylline. FAS-APO1, which is a marker of apoptosis, cell suicide, fell with pentoxifylline. BNP, B-type natriuretic protein, this is a protein released by the heart in response to stress or strain. The higher the level of BNP, the worse your heart failure. It rose with placebo therapy. It fell dramatically from 267 to 46 with pentoxifylline. By blunting deleterious overexpressed levels of TNF and other inflammatory cytokines with pentoxifylline, we reduce the levels of these inflammatory mediators. We reduce the rate at which the heart cells commit suicide, apoptosis. The heart performs better, BNP levels fall, treadmill time fell with placebo therapy, it rose with pentoxifylline, New York Heart Association functional class fell, that's an improvement more with pentoxifylline. Ejection fraction rose with pentoxifylline from 28 to 37%. 10 of the 19 patients treated with pentoxifylline increased their ejection fraction by over 10 points, which was highly significant. Now that's a benefit of pentoxifylline in heart failure due to coronary disease. What about non-coronary autoimmune dilated cardiomyopathy? Will pentoxifylline help there? 39 patients with heart failure due to dilated cardiomyopathy all had an ejection fraction below 40%, and they're all on standard drug therapy. After 3 months of observation, you randomize them to receive pentoxifylline, 400 milligrams 3 times a day, or placebo. Otherwise, same care, drug therapy is not altered, reevaluate at 6 months, 5 died, 3 in the placebo group, and you're going to look at the rest. And what you find, there's very little effect on TNF-alpha in this study. FAS-APO1 did not change with placebo therapy. It fell with pentoxifylline. Ejection fraction rose from 24 to 31% with pentoxifylline, treadmill time improved. Another positive functional result. Functional class, how well you're doing, improved in 67% of those on pentoxifylline versus only 10% on placebo. So in heart failure due to coronary disease, heart failure due to cardiomyopathy, cytokine modulation with pentoxifylline is effective. These studies have been carried out in different causes of heart failure. Hypertensive cardiomyopathy, postpartum cardiomyopathy, that's the cardiomyopathy of pregnancy. In all these models of heart failure, it doesn't really matter the cause because all heart failure is associated with a Th1, Th17 imbalance. We see improvements in symptomatic status, ejection fraction, lab markers, and outcome. So because all heart failure is associated with a maladaptive immune response, pentoxifylline is of universal value in the treatment of heart failure in humans. Let's look at pentoxifylline in experimental atherosclerosis. If it benefiting the immune system in heart failure, would it also be helpful in atherosclerosis? 8-week-old ApoE double knockout mice. Now these mice are going to develop atherosclerosis on a standard diet. You'll treat them with daily injections of pentoxifylline or vehicle control substance. You're going to sacrifice them at 4 and 12 weeks, evaluate lab, and look at how much plaque is present in the artery wall. Now, lipid values at 12 weeks, cholesterol is high in both groups, and that's because these are genetically abnormal mice that have a high cholesterol level independent of diet. And there was really no effect on HDL. Triglycerides were a little bit lower in pentoxifylline. So pentoxifylline didn't have a major lipid effect. If we look at VCAM1, that is an adhesion molecule expressed by the endothelial cells to pull in white cells, that's a marker of inflammation, it was attenuated in the animals receiving pentoxifylline. If we look at the TH bias, the percentage of T cells expressing interferon gamma, Th1 cells, the pentoxifylline animals had lower levels of Th1 than did the untreated animals. If we look at the severity of the inflammatory infiltrate into the plaque, we see lower levels of immune cells in the animals treated with pentoxifylline. If we look at percentage of the arterial wall covered with plaque, at 4 weeks there was no real protection with pentoxifylline. However, at 12 weeks, 5.5% of the artery wall was covered with plaque in the untreated animals with genetic hyperlipidemia, in the animals with genetic hyperlipidemia, predisposed to atherosclerosis. By modulating the immune response to the hyperlipidemia with pentoxifylline, there was much less plaque. Well, what about in humans? Here's a study of pentoxifylline in hypertensive diabetics. Hypertension and diabetes increase your risk of cardiovascular disease and inflammation. Can we modulate that with pentoxifylline? 51 hypertensive type 2 diabetics, all at least 45 years of age, all on metformin, that is a drug that promotes insulin sensitivity at the level of the liver. And they were also on sulfonylurea agents. Those are agents that encourage the pancreas to kick out more insulin. All on antihypertensive drug therapy, but none were on immune modulating drugs such as statins, ACE inhibitors, or angiotensin receptor blockers. Baseline lab study randomized them to receive pentoxifylline, 400 milligrams 3 times a year placebo. All their prior meds were continued. Baseline, you repeat the baseline studies at 4 weeks. A double-blind protocol was followed. Fasting blood sugar fell in both groups. It fell a little bit more with pentoxifylline. Hemoglobin A1C, which is a lab marker of your average degree of blood sugar control over 3 months, fell in both groups. It fell a little bit more with pentoxifylline. By blunting inflammation, you have less insulin insensitivity. C-reactive protein fell with pentoxifylline. White blood count fell with pentoxifylline. Serum albumin rose. Albumin is made by the liver, and you want a high level of albumin. Under inflammatory stress, the liver's not functioning as well. It doesn't make as much albumin. By blunting inflammation with pentoxifylline, the liver functions better. You have better insulin sensitivity, better liver synthetic function, so you make more albumin. Malondialdehyde is a marker of free radical stress. We showed that ECP lowers malondialdehyde. Antioxidants lower malondialdehyde. So does pentoxifylline by blunting inflammation. Glutathione levels rose. That is our key antioxidant. We showed in the Lupus study that fish oil is associated with a rise in the levels of this key intracellular antioxidant, glutathione. The same happens with pentoxifylline. And if their predicted 10-year event risk was unchanged with placebo therapy, it decreased with pentoxifylline due to its anti-inflammatory effect. Okay, so pentoxifylline is a value in animal models and in risk factor reduction. What effect will pentoxifylline have in patients with established cardiovascular disease? First, let's look at 11 men with chronic stable angina, all with an at least 70% narrowing in one artery, 5 with prior coronary bypass surgery. You do a baseline evaluation, assess the severity of their symptoms, how many nitros they needed, how often they had angina. You do a baseline stress EKG study as an objective measure of their ischemic burden. Begin pentoxifylline, all prior meds continued, repeat the baseline studies at 6 weeks. Reevaluate at 6 weeks. Now, clinically, they improved. 9 of the 11 men noted a reduction in angina frequency. The 2 who did not improve had severe inoperable 3-vessel disease. So their symptoms improved with pentoxifylline. Treadmill parameters increased significantly. Treadmill time increased from 7.7 to 10.1 minutes. Time to chest pain increased from 5.5 to 7.9 minutes. Time to ST depression, that's the EKG marker that the heart muscle is not getting enough oxygenated blood, also improved. So symptoms improved and treadmill parameters improved by modulating the Th1, Th17 immune dysregulation associated with their stable atherosclerosis. What about unstable angina? We know that unstable angina is associated with an immune frenzy. What effect will pentoxifylline have? 64 patients presenting with unstable angina or a non-Q wave, that's a non-full thickness myocardial infarction, heart attack. Do baseline lab studies. They get their standard medical therapy for acute coronary syndrome, and they receive an angiogram, and if needed, a balloon angioplasty or stent on day 1 or 2. You're going to discharge them all on standard medical therapy, including as tolerated ACE inhibitors, beta blockers, and a statin. Then you randomize them to receive, on top of standard medical therapy, either placebo or pentoxifylline 400 milligrams 3 times a day, reevaluate at 1 and 6 months. CRP fell in both groups. The farther you get away from the acute event, you would expect inflammation to quiet down. You'd see a reduction in CRP, but the nadir level of CRP fell more with pentoxifylline, as you would expect. Interleukin-6 fell in both groups a little bit more with pentoxifylline. Tumor necrosis factor alpha didn't change much in either group over time. Interferon gamma fell a little bit more with pentoxifylline. So lab markers of Th1, Th17 activation fell as you got away from the event. They fell a little bit more if you modulate the immune system with pentoxifylline. Those numbers themselves are not very impressive. Transforming growth factor beta rose in both groups. IL-10 fell in both groups. Again, in response to this inflammation, you'll have a Treg response that then tails off. It doesn't tail off as much with pentoxifylline. So pentoxifylline kind of downregulates Th1, Th17, upregulates Treg. Now, what was very impressive, though, was 6-month event rate, recurrent event. 34% in placebo, 13% with pentoxifylline. Which group would you want to be in? If you had an acute coronary event and I give you pentoxifylline, I'm going to provide a great deal of protection against recurrent events. Why? Recurrent events are due to an immune frenzy. If we prevent immune frenzy by downregulating with pentoxifylline, far fewer events. So it works in stable angina, it works in unstable angina or non-Q wave infarction. What about bypass grafts? You know, when we put in bypass grafts, they fill up with atherosclerosis much more rapidly than did your native arteries, and that's because veins don't make much nitric oxide. Will pentoxifylline protect your grafts from atherosclerosis? Here we're going to look at 212 patients undergoing vein graft revascularization. 300 were undergoing coronary artery bypass surgery, 110 received vein grafts for peripheral vascular disease. You randomize them to standard care or pentoxifylline 400 milligrams intraoperatively in the priming fluid or into the artery being bypassed in those with peripheral vascular disease, then 400 milligrams daily over 5 years. You'll evaluate them before and at 1, 6, 12, and 24 months following surgery. Now, if you had coronary bypass surgery, they would do a stress test or perfusion scan and then an angiogram if that was abnormal to look for graft failure. If it was peripheral vascular surgery that you received, they would do a Doppler study and then an angiogram if that was abnormal to document patency or failure of your peripheral vein grafts. What you find of those that underwent coronary bypass graft surgery, at 1 month, 94% of those on placebo therapy were patent, 98% on pentoxifylline. As you go 6, 12, 24 months, in both groups, you see an increased rate of graft failure, but there was less graft failure in the patients receiving pentoxifylline. At 24 months, 81% of the grafts in the placebo patients were still patent versus 93% with pentoxifylline, a significant protection. Same situation with peripheral vein grafts. At 2 years, 96% of those on pentoxifylline, their grafts remained open versus only 78% with control therapy. So at all these phases of atherosclerosis, just as in all causes of heart failure, modulating the immune response with pentoxifylline provides clinical benefit. Pentoxifylline in chronic kidney disease. Many of the drugs that are good for the heart are bad for the kidney. Many drugs that are good for the kidney are bad for the heart. What effect will pentoxifylline have? Well, I can tell you that chronic kidney disease is also associated with Th1, Th17 upregulation, a reduction in TGF. Free radicals will damage the vascular cells of the kidney just as they damage the vascular cells in the heart, causing autoimmune attack on the kidney. Well, pentoxifylline blunts autoimmune damage in the heart. Will it work in the kidney? 85 patients with chronic kidney disease. Their glomerular filtration rate, it should be 100. Theirs is impaired. It's between 10 and 60. All are spilling protein. The sieve was leaky, and protein is leaving their blood going out in the urine. They're all on losartan, which is an angiotensin receptor blocker. That is a beneficial drug. It quiets down Th1, Th17 within the artery wall. It does the same thing in the kidney, so it's protective. Do a baseline lab study, then randomize them to receive pentoxifylline 400 milligrams, 1 to 2 doses per day on top of losartan or losartan and placebo. Other treatments are held constant. Reevaluate at 12 months. At that point, all those who were on placebo therapy are crossed over to pentoxifylline because they had demonstrated that pentoxifylline was of value. If we look at the glomerular filtration rate, it decreased in both groups, but it decreased slower in those on pentoxifylline. Proteinuria decreased by 24% in those on pentoxifylline. It increased on placebo therapy. Here, if we look at the degree of protein spillage, it's increasing over 12 months with placebo therapy. Then you cross them over to pentoxifylline, it decreases. So in those that were on placebo and then went to pentoxifylline, at first they're leaking more protein and then the sieve is less leaky on pentoxifylline. Glomerular filtration rate declined by 2.4 points per year with placebo therapy, much less with pentoxifylline. So deterioration in chronic kidney function is attenuated with immune modulation with pentoxifylline. Now, if we look at urine tumor necrosis factor alpha, a Th1, Th17 cytokine, it's decreased with pentoxifylline. Urine MCP1, monocyte chemotactic protein, released by the inflamed kidney to call in more immune cells from the bone marrow, it is also decreased with pentoxifylline. So the cardiovascular benefits are also holding in the kidney. So Pentoxifylline is of universal value in cardiovascular disease and presumably all other Th1, Th17 conditions. It shifts the immune bias from Th1 to Th2 and Treg in animals and humans, decreases interferon gamma, TNF-alpha, interleukin-1 beta, interleukin-6, interleukin-8, while increasing the Treg cytokines, IL-10 and transforming growth factor beta, reduces Th1 inflammation, white cell adhesion molecule elaboration, and lesion size in animals predisposed to atherosclerosis, individuals with diabetes and hypertension. It lowers risk factors and inflammatory mediators. Treadmill parameters improve in patients with stable angina. In patients that had an acute coronary syndrome, we see an improvement in biochemistry and dramatic improvement in outcome. It helps prevent atherosclerosis in vein grafts. They're less likely to close down. It decreases apoptosis in our heart cells, so the heart failure is slowed down. Improvement in functional status, ejection fraction, outcome in heart failure of any cause, decreases proteinuria and functional decline in chronic kidney disease. And I didn't present these studies. It decreases mortality in sepsis, helps in fatty liver by shifting away from Th1, Th17 towards Th2 and a Treg physiology. Famotidine. Now, this is the generic name for Pepcid, an over-the-counter agent that blocks the histamine 2 receptor in the stomach. The histamine 2 receptor in the stomach makes acid. So if you have stomach acid problems, you might take Pepcid or Famotidine to blunt stomach acidity, and that will make you feel better. 20 years ago, when I was spending all my time in the hospital, we would be troubled by patients with stress gastritis. Due to the stress of their heart attack or heart failure, they're making a lot of stomach acid, they'd have GI bleeds. So we would give you intravenous Famotidine to quiet that down. I came across a paper showing that Famotidine lowers ejection fraction when it is abnormally elevated. So it has an effect on the heart. And I thought about that, was this a good thing or a bad thing? But it didn't seem to be causing heart failure, so I kind of filed that away. And now we understand how Famotidine might work in heart failure, because the failing heart upregulates a histamine receptor. And that plays a role in ongoing damage to the heart. 50 patients with symptomatic heart failure, and they had reflux. Their ejection fraction is below 30%. They're on standard therapy with diuretics, beta blockers, ACE inhibitors, spironolactone. And they were still having symptoms of congestive heart failure despite standard neuroendocrine blockade. Do a baseline evaluation, randomize them to receive Famotidine, which is Pepcid, or a control agent called Tepranone. This study was done in Japan. This is a Japanese drug that doesn't have anything to do with the histamine receptor. Otherwise, the same care, drug therapy is not changed. Reevaluated at 6 months, double-blind protocol was followed. And what you find, if you do the cardiac echoes, the chamber size decreases with Famotidine therapy. The left atrium decreases in size. The degree of pulmonary hypertension decreases. So their hearts get smaller because the degree of heart failure is improving with Pepcid. If we look at BNP, B-type natriuretic peptide, nothing happens with control in the control group that are receiving maximal medical therapy. But if you add Pepcid to block the histamine receptor, if you add that to maximal medical therapy, you see a nice reduction in BNP. Hospitalization for an exacerbation of heart failure over 6 months falls from 24% to 4%. Wow, that is fantastic. We use beta blockers to block the effect of adrenaline and noradrenaline on the heart. These catecholamines whip the heart, and they basically whip the heart to death. That's why we use beta blockers in heart failure. The failing heart will also upregulate its histamine receptor. It ties into the beta receptor. So by blocking this upregulated histamine receptor, we can prevent the heart basically from destroying itself. So by adding Pepcid, which is now over the counter, to standard drug therapy, we get biochemical improvements and an improvement in outcome. And this is a universal and inexpensive therapy for heart failure. No one yet has done studies of histamine blockade in arterial disease. However, Mast cells are allergic cells that release histamine. We see a lot of histamine in unstable plaques. So perhaps studies in the future will show that famotidine is helpful in arterial disease. It's definitely helpful in congestive heart failure. Carvedilol is a beta blocker that also has an antioxidant effect. It is our favorite beta blocker in the treatment of heart failure and in individuals who are overweight and diabetic, because beta blockers tend to promote accumulation of fat in the viscera and insulin insensitivity. Carvedilol does not. Its antioxidant effect provides an incremental better effect in the treatment of heart failure than do other beta blockers. So does it have an immune effect? 60 patients with symptomatic pump dysfunction, ejection fraction below 40%, 35, the heart failure was due to coronary disease, 25, it was non-coronary dilated cardiomyopathy. All 60 are receiving standard heart failure therapy with digoxin, a diuretic furosemide, and an enalapril, that is an ACE inhibitor that does not get into the heart muscle or the artery wall. And you add carvedilol, a beta blocker with additional vasodilatory and antioxidant effects, to these patients, and you see an improvement in functional class and ejection fraction as we would expect. TNF levels rise as the severity of heart failure increases, as do IL-6 levels. And what you find with standard therapy, over time we see a rise in IL-2 that causes T cells to proliferate that is blunted by carvedilol. We see a reduction in TNF, a reduction in IL-6. So carvedilol, above and beyond standard beta blocker therapy, also has immune modulating benefits. Many of the drugs we use in cardiovascular disease that work, statins, ACE inhibitors, carvedilol, work at least in part by decreasing the maladaptive immune bias, too much Th1, too much Th17, not enough Treg, that characterizes atherosclerosis and heart failure. Now, we'll conclude our presentation with a summary and a plan of action in the next section. This section summarizes our discussion on immune mechanisms in cardiovascular disease and initiates a plan of action. At the beginning of this presentation, I asked for the privilege of trying to convince you that atherosclerosis and heart failure at heart are due to an imbalanced immune response to perceived infection. LDL cholesterol infiltrates the artery wall. It is altered, trapped, and oxidized by free radicals. It is then misrecognized by immune cells, macrophages, as an invader. The macrophage gobbles up the LDL, breaks it up into little pieces, initiates an immune response, calls in other immune cell reinforcements. It will then present a little snippet of that oxidized LDL to a resting T cell that will activate, release cytokines, and promote progression of atherosclerosis and heart failure. Our immune response is unbalanced because we are overweight, because we have sleep apnea, because we have a high saturated fat diet, because our diet is low in minerals. We tend to have an imbalance with too much Th1, too much 17, not enough Treg. So we have an unbalanced immune response to perceived infection of the artery wall with oxidized LDL, and thus over time our arteries fill up with lipids, fill up with macrophages, and then in an immune frenzy, the fibrous cap is ruptured and we have an acute coronary event, a heart attack or unstable angina. Similarly, heart failure is due to a maladaptive Th1, Th17 low Treg response to perceived infection of the heart with heart proteins that are exposed inappropriately to the circulation. We showed how immunizing a mouse against troponin I will lead to experimental heart failure or a large experimental heart attack. And conversely, if we can generate a Treg response to troponin I, we can protect the animal from heart failure. In human heart failure of any cause, be it cardiomyopathy, valvular disease, hypertensive heart disease, or coronary disease, there is always a maladaptive response to the immune system to perceived infection of the heart with heart proteins that have been exposed to the circulation. Now, heart failure and atherosclerosis are associated with an imbalance. We don't have enough Treg cells, we have too many Th1 and Th17 cells, and that also leads to an imbalance in cytokine levels in the circulation, within the artery wall, and within the heart muscle itself. What are the antigenic determinants in the artery wall and in the heart that the immune system is inappropriately responding to? We know in the artery wall, this is begun with oxidized LDL and oxidized LPA fragments, heat shock protein, beta-2 glycoprotein, and proteins from bacteria and viruses that hitchhike into the artery wall on white cells that gobble them up elsewhere, but which are called in as reinforcements to deal with this perceived infection with oxidized LDL in the artery wall. In the heart muscle, we know that troponin I is antigenic, so is myosin. There may be other proteins that are antigenic. How do we treat immune dysregulation in heart failure, arterial disease? Well, in the old days, they used cytotoxic therapy or antibody immunoabsorption, but what we're going to do is we're going to carry out milieu therapy. We're going to convert you from this Th1, Th17 bias to a Treg bias, and we covered studies showing that pharmaceutical intervention with pentoxifylline, statins, angiotensin II receptor blockers, or drugs that block the formation of angiotensin II downregulate the immune response, thus they're of value in atherosclerosis and in heart failure. And nutritionally, substances such as vitamin D, fish oil, weight loss, antioxidants, testosterone, metal detoxification do the same thing. We can also neutralize the maladaptive T cell response to specific antigenic determinants. We could administer false targets to which activated T cells will bind to and be neutralized. We can also administer, and we've demonstrated this in animal studies, antigenic determinants to the animal in a non-inflammatory Treg milieu. Thus, the animal will mount a Treg response to the antigenic determinant, and that would tend to tamp down any Th1, Th17 deleterious activity in the organ that might be subject to autoimmune attack. What's the plan of action here? How do we avoid atherosclerosis? Now, that's a long discussion. Here I'm going to talk about how you can avoid the autoimmune components of atherosclerosis. Well, do not allow oxidation, glycation, or alteration of LDL within the artery wall. All right? Maintain a low LDL and lipoprotein level, and of course, block oxidation, alteration, glycation, and inflammation. Maintain a high Treg and a low Th1, Th17 immune bias, and we've discussed how to do that. Same thing with heart failure. Do not allow myocardial proteins to enter the circulation. Don't have a heart attack. If you have high blood pressure, lower it. If you're born with an abnormal valve, I can't do anything about that, although I can modulate the immune response. How to avoid autoimmune disease in general? Avoid damage to self proteins. Don't take in toxins that will alter self. And again, maintain a Treg high, low Th1, Th17 milieu. Anything that's beneficial in atherosclerosis and heart failure is going to be beneficial in autoimmune disease in general. We demonstrated that with fish oil and vitamin D and testosterone. More specifically, lipid management. I want you to have an LDL cholesterol level appropriate to your health condition. If you're 20 years old and your cholesterol is slightly high, but you do not have mercury fillings, you only eat organic food, you're a trim athlete, and you live out in the countryside where there's no toxicity, as close to the theoretical Garden of Eden as possible, I know that your LDL is not going to be oxidized, and I don't need to lower your LDL because, again, LDL only hurts us when it is oxidized, glycated, or inflamed. Conversely, if you're 70 years old, you've had 2 open heart surgeries, you just had an episode of unstable angina, and your C-reactive protein is off the chart, I know that you are subject to a Th1, Th17 immune storm, you have no Treg cells, any LDL that you have is immediately going to be oxidized, then I'm going to use nutrition, drugs, and diet. I want your LDL to go down to 50 because I know that any LDL is going to be oxidized. So the ideal LDL is a function of your overall health and state of activation of your immune system. Now, one thing that everybody can do is lower your body burden of infection, toxic metals, and inflammation. We showed that any infection, any inflammation, and heavy metal overload makes your cholesterol go up. So get rid of those things. That's good for your cardiovascular health. It's good for your health in general. And if we do need to lower your LDL, we can use dietary, nutritional, and pharmaceutical management. Maintain a healthy endothelial surface. If the endothelial cells are healthy, they make a lot of nitric oxide. They kind of resist oxidation and inflammation. They will not elaborate as many adhesion molecules. That'll keep the monocytes from entering the artery wall, becoming macrophages to gobble up the oxidized LDL. Exercise on a regular basis, regular laminar shear stress, as we achieve with the ECP, you can achieve with exercise, and downregulate the immune response in the artery wall. The higher your blood pressure, the greater is the filtration force to get the LDL particles into the artery wall. We want a healthy blood pressure. Oxidant-antioxidant balance is key. Again, if your antioxidant defenses are strong, you will not oxidize your LDL. Unfortunately, our diet is weak in antioxidant, vitamin, and minerals, and we are all overloaded with toxic metals that Mother Nature never planned on, so we're subject to free radical stress. Free radical oxidative stress in general promotes Th1, Th17, lowers Treg. So supplement with antioxidant vitamins, minerals, bioflavonoids, get the toxic metals out of your body, and minimize non-cardiac inflammation. We want to see an ideal weight. 2/3 of adult Americans are overweight or obese. We have a BMI of 25 or above, and even that is not ideal. We want to get your BMI down to 22 or 23, get rid of visceral fat, get rid of this Th1 inflammation, and control sleep pattern. If you lose weight, your sleep pattern will go away. Chronic infections, particularly gingivitis, cause chronic inflammation. We showed that as the severity of your gingivitis increases, your CRP rises. That's not good for us. Deal with chronic inflammation and normalize your blood sugar, because the LDL that gets glycated is the LDL that will initiate the immune response, just like oxidized LDL. Some general principles. Now, some of you have specific health conditions that require specific diets. Here I'm just going to be talking in general principles. Eat organic as much as possible. We don't want pesticides and herbicides in our food. Mother Nature never planned on this. Particularly do your best to go organic with meat, because non-organic meat has 20 times the level of pesticides as does non-organic fruits and vegetables. So as much as possible, eat organic. Eat, as my wife would say, as close to the earth as possible. The less processing of your food, the better. We've taken food from the earth and we mill it and we grind it and we package it. And by the time we eat it, it's very different from the nutritious material that the earth gave us. White bread and white sugar are raw materials that have been processed. All the good nutrients leave. All we get are the inflammatory materials. Here's an interesting story. Stanley and Livingstone penetrated the interior of Africa in the late 1800s. They were followed by missionaries and missionary hospitals. And in 1900, a paper was published of diseases of these new people, the Africans. And they saw a lot of broken bones. They saw a lot of trauma. They saw infections. But in 1900, they saw no hypertension, no diabetes, no osteoporosis, no appendicitis, and no cancer. And it was felt that Africans were genetically protected against these diseases. They were genetically protected against cancer. Then in 1903, an African man developed a cancer. And this created a big stir in European medical circles because this was felt to be impossible. Turns out this man was working at a mining company, and he was living with and eating the same foods as the Europeans, a processed inflammatory diet. Well, over the years, Africans shifted from eating close to the earth and began to adopt European principles of processing their food, and now African blacks are as sick as we European Caucasians are. But if you eat real food the way Mother Nature intended, we don't get all these diseases. Another point if your ancestors originated in another region of the earth, in another society, you really ought to be eating the foods indigenous to your ancestral homeland, at least the foods that people ate over 200 years ago before we started processing foods. Our genes adapt to optimize our chance of survival. Thus, the best diet for a Northern European may not be the best diet for a Southern European, vice versa. When Japanese move to the United States, they get sick really quickly because they're eating these processed American foods, not the Japanese foods that they are genetically best suited for. So kind of eat close to the earth and think about what type of foods you were designed to eat. For Caucasians from Europe, a Southern European Mediterranean diet is best. Plenty of pigmented fruits and vegetables, olive oil instead of butter, fish and poultry with less red meat, and essential fatty acid-rich nuts. Exercise on a regular basis. All of you have excuses as to why you can't exercise. You're being foolish. Find some way to exercise. It's critical to maintaining good health. It's critical to maintaining a balanced immune system. Find an exercise program that you enjoy and get hooked to the point where it's part of your life. You want to be an athlete to some degree until the day you die. Look at the causes of stress in your life and deal with them. Try your best to enrich your family life and pay attention to your spiritual life. These are all important in maintaining good health. Supplements. Now, we will construct supplement programs unique to an individual based upon my evaluation of you, but in general principles, I recommend a broad spectrum 6-tablet-a-day vitamin and mineral. A 1-a-day does not give you all the good stuff that you need. So if you're going to take a 6-a-day vitamin and mineral, take 2 with each meal to block inflammation related to the foods you eat. Additional specific supplements have indicated by testing. Everybody ought to be taking essential fatty acids and vitamin D, 1 to 3 teaspoons of fish oil, and that's the omega-3. And we want a little bit of the beneficial omega-6, gamma-linolenic acid from evening primrose oil or borage oil. Everybody ought to be taking, it's very unusual to see someone native to Northwest Ohio that does not have a low vitamin D level. So we measure vitamin D and we're going to supplement you and get a mid-range, mid-normal range level. And the typical individual in the Midwest needs 2,000 to 6,000 international units a day. If you have any degree of arterial disease, if you have atherosclerosis, hypertension, heart failure, or an undesirable carotid IMT, supplement with arginine, 2,000 to 4,000 milligrams 3 times a day. This is the raw material the endothelial cells pick up and use to generate nitric oxide. Lipid management. If I want to lower your lipids pharmacologically, I'm going to use statins because they lower your LDL and they downregulate the immune response. Green statins, such as Reggie's rice extract and polycosanol, will lower your LDL, not as much as the pharmaceutical statins, and they also have immune modulation effects. Statins waste testosterone and coenzyme Q, so I will supplement men with, if I give you a statin, I will measure your testosterone. If it falls, I'll give you testosterone. Both men and women who receive statins should be taking coenzyme Q. If my goal is to raise your HDL, I will use niacin and or fish oil. If you have a high LPA, I can't lower it, it's under genetic control, but I'll suggest that you avoid trans fats, that you supplement with fish oils, and we'll bind it up with lysine proline so it can't enter the artery wall, and will increase the resiliency of the artery wall with vitamin C to keep the LPA out. If I need to lower your blood pressure, I'm going to use an agent that has beneficial immune effects, and that would be the tissue-specific ACE inhibitors, quinapril or ramapil, or an angiotensin receptor blocker will be agents of first choice. Second, I will use carvedilol because in contrast to other beta blockers, it has an antioxidant and a beneficial immune effect. Other antihypertensive agents such as diuretics, calcium channel blockers, or alpha blockers do not have immune benefits. They really aren't as helpful. If you have atherosclerosis, I'm going to treat you with a combination of nutritional supplements and pharmacologic intervention with an aggressiveness commensurate to how much trouble you're in. How extensive is your disease? How inflamed are you? What is your risk of getting into trouble? And I will use statin therapy as tolerated. Now, I don't push for max doses. In standard drug medicine, when the only approach to immune modulation is really statins, they want to push to get the LDL as low as possible. I want to lower your LDL, but I'm also concerned about side effects. And given that I have more than one hammer to use, I can use lower doses of statins, lower your LDL, but because I'm also intervening with all these other pharmacologic and nutritional immune modulators. All my patients with atherosclerosis, unless your blood pressure is low, I feel you would benefit from an angiotensin receptor blocker or a tissue-specific ACE inhibitor. In the studies that we presented showing benefits of these agents, we weren't treating hypertension, we were treating Th1, Th17 inflammation. In men, it is rare for me to see a new male patient with cardiovascular disease that does not have a low normal testosterone. And because if they had high testosterone levels, they probably wouldn't have atherosclerosis. So I will supplement with testosterone to achieve a high normal testosterone level to obtain a steady level. I use testosterone replacement like I use statins. I want a steady level. We use pellet testosterone replacement to provide the steadiest levels over time. Both men and women will benefit from pentoxifylline. This drug is off patent, it's inexpensive. We can use enhanced external counterpulsation, not just to treat your symptoms by promoting collateralization, but it also improves your outcome by supporting the endothelium and, as we demonstrated, down-regulating Th1, Th17 immune function. If you have heart failure, you're going to receive a tissue-specific ACE inhibitor or an angiotensin receptor blocker unless your blood pressure is too low. I will give you carvedilol unless your blood pressure, heart rate are too low. Spironolactone does not have immune effects, but we use it in all of our patients because it blocks a deleterious hormone called aldosterone that makes the heart stiff. I will use pentoxifylline in any cause heart failure. In men, I will use testosterone, preferably with pellet replacement, and I will use low-dose statin therapy. Higher doses of statin therapies may produce incremental benefits in atherosclerosis. So far, the literature on statin therapy and heart failure, low doses, say 10 milligrams of Lipitor, atorvastatin, 10 to 20 milligrams of Zocor, simvastatin, will provide benefit. And of course, statins will waste coenzyme Q, so we always supplement with CoQ. We can use external counterpulsation in heart failure. We typically use it in angina, coronary insufficiency, but it is a value in heart failure. We talked briefly about MME, magnetic molecular energizer therapy. I presented one patient whose cardiomyopathy improved rapidly with MME. We get the metals out, less oxidative stress, and of course, formal immune modulation, the therapy we used in the case studies that I presented to you. Working with us. If you want to work with us at Comprehensive Heart Care in Toledo, the first thing I want you to do is learn all you can about your health and your disease conditions and about the treatments available. Towards that end, you can read Reverse Heart Disease Now that I wrote with my colleague Stephen Sinatra. Now, the book's 4 years old. We don't talk a lot about immune modulation, but it tells you a lot about our philosophy, our integrated philosophy to cardiovascular disease. We have learning tools available. DVD presentations are available on our website, or you can obtain them from the office, mostly on DVDs, and we have a few older talks that are available only on cassette tapes. If you still have a cassette player and you give me the cassette tapes, we'll copy them for you so you can learn about these other topics. Now, my medical practice is called Comprehensive Heart Care, and it's been in place for 25 years. And over the years, we brought in additional therapies. So we also have the ECP Center of Northwest Ohio, Total Body Balance Medical Weight Loss Center. If we help you lose weight, we can lower inflammation. The Advanced Magnetic Research Institute of Northwest Ohio, and we're involved in a number of clinical research studies. So some patients are referred to me for ECP or MME or for weight loss. But if you're interested in immune modulation, you'll be a patient within our integrative cardiology practice. Now, if you want to work with us, we are happy to work with you, but you need to understand that my practice may be a little different from other medical practices with which you've been affiliated. I love what I do. I get up in the morning, I go to work, and all of a sudden it's time to go home, and I don't know where the time went because I'm actually having fun. And that's because I'm working with patients who are engaged. If you want to work with me, it's necessary that you become engaged in the process. If you simply want me to tell you what to do, you're not going to be happy working with me, and I won't be happy working with you. My patients push me to learn more. I push my patients to learn more. We actually, kind of have fun working together. My staff enjoys working in this process, and this is the way I want to practice medicine. And so your engagement is critical. Another point, I work only for you. Now that sounds like a silly thing for a doctor to say, but it is very important. As a younger doctor, I thought I was doing my best, but in retrospect, what I was really doing was meeting the needs and expectations of the hospital, other doctors, drug companies, etc. I was doing my best. This was what I was taught to do, but really, I wasn't putting your outcome first. Now that I'm a middle-aged doctor, I have the luxury of doing things simply because they're the right thing to do. So I'm going to put you first. Many of my recommendations that I think are in your best interest may not sit well with the hospitals or the insurance companies or your other doctors. And at this point, I don't really care. I don't want to antagonize anybody. I don't want to pick a fight. But if I think a therapy is in your best interest, this is what I'm going to recommend. If you want a doctor who's going to please other doctors and strive for consensus, that's not me. I don't compromise, or people have told me I don't compromise very well. I'm not really interested at this point in my career in doing anything but my best. So if you want me to knock myself out to help you, I'm happy to do that, and I'm going to ask you to knock yourself out, making it easy for me to help you. Your cooperation is critical. Now, I don't go to the hospital anymore. I used to spend all my time in the hospital. Then I got involved in enhanced external counterpulsation 15 years ago. We were the 22nd practice in the United States and the second in the state. We are the only ECP program within 200 miles. So soon we had a very vigorous program. At one point we had the busiest ECP program in the United States. From dawn to dusk we were doing ECP, and this was a lot of fun. I was helping a lot of people. Then Medicare came out with this rule and said the doctor has to be there the whole time. Well, that put me in an impossible situation. If I had to leave the office to go to the hospital and handle an emergency, then I was in violation of Medicare policy. So what I was doing is getting up early and going to the hospital, or going to the hospital at night, and this was exhausting. Plus, you really can't provide good care for hospital patients over the phone all the time. So to solve this problem, I began to give up reading nuclear studies, give up reading echoes, and then give up management of my patients in the hospital. And how this works, I don't go to the hospital, but I keep my beeper on. If there's a problem, I get a call. If it can be solved over the phone, I solve it. If you need to be admitted, you will be admitted to a colleague who will look after you in the hospital. I talk to that colleague, I send your records, so there's continuity of care. And the doctors who work with me in the hospital are very, very good. They focus on hospital medicine, and they're not anti-nutritional medicine. So if you want a doctor that will see you in the office and in the hospital, that's not me. I've chosen to confine myself to the office. Now, because I'm not going to the hospital anymore and I'm in the office, I'm pretty much always on time. So I'm going to ask you to be on time. If you come in 20 minutes late, don't expect me to see you, because then you would put me 20 minutes behind for all the other patients. That would not be fair to them. If you're supposed to get lab work done before you see me, so I have the lab in hand when I see you, please do that. If I see you and the lab comes in the next day, that's just a lot of extra work for me, and it's not in your best interest. So I want to knock myself out to help you and keep you out of trouble. I'm going to ask you to knock yourself out to make it easy to achieve this. Now, I practice integrative medicine. I resist this polarization into alternative and conventional medicine. I think that's silly. So I will use drug therapy when I think it's appropriate. I will use nutritional therapy when I think it's appropriate. I will utilize detoxification therapy when I think it's appropriate. And I'm not really worried about standard of care versus non-standard of care. I think that there's no such thing as conventional medicine or alternative medicine. I think it's just medicine, and it's integrative medicine to take the best of standard therapy, the best of nutritional therapy to get the best results for you. If you're interested in this approach, give us a call. You can look at the website, heartfixer.com. You can read the book. You can give us a call at 419-531-4235. If you live out of town, a good idea is to do a phone consult. I get your records, I think about them, I talk with you, then we can plan your visit to Toledo, therefore your time is better spent. Some of the diagnostic studies that we do would require you to cut back on some of your medicines, or some of the other studies would require you to supplement with some materials before you come to Toledo, so we can make your visit in Toledo more efficient with a phone consult ahead of time. So if you're interested in this, give us a call. And remember, one thing about our practice, the inflammation stops here because we're looking after your T cells. Thank you very much.